Ladies and gentlemen, good morning. I'm Jim Byron, President and CEO of the Richard Nixon Foundation. Welcome back to the Nixon Library as we start day 2 of the Nixon National Cancer Conference. I want to thank our board members that are here in the room with us, and I want to also thank the NCI Cancer Center directors. There are at least 5 running around here somewhere who have flown in from all over the country, so thank you very much for being here. Last evening, we were treated to a rousing discussion between industry leaders about the mathematical underpinnings of cancer and how and why we understand cancer today as we do, and how that can help scientists to develop a cure in due time. And last evening, in a video from 1972, we heard President Nixon say that you cannot separate one disease out without having experts in other fields making contributions. So we're going to cover a host of topics today in keeping with that theme. We begin this morning with a program on regenerative medicine and stem cell therapy, perhaps one of the most interesting and rapidly evolving aspects of modern medicine. And here to lead this program is Dr. Donald Skip Trump, founding CEO of the Inova Shar Cancer Institute and Inova Health Systems, and a professor of medicine at the University of Virginia Cancer Center. He is also the co-author of the new book, Centers of the Cancer Universe, A Half-Century of Progress Against Cancer, which I will add is for sale in the Nixon Library's gift shop. Doctor? Okay.
Well, thanks very much for the kind introduction. I'll try to restrain myself from advocating that you stop in the bookshop and buy a book. I'm really delighted to be here, and most delighted to be here with a panel of distinguished colleagues. This is something I've been looking forward to, and hopefully we'll have a discussion this morning that really does outline where we started, where we've been, and what the future might hold, with a particular emphasis on cellular therapies. In keeping with the historical theme that this conference embodies, I think it's interesting to reflect on the history of cellular therapy. In 1957, Don Thomas in the New England Journal reported the first stem cell infusion in humans. 6 patients were treated after chemotherapy and total body radiation. 2 patients had a transient graft. They all died, but that established the feasibility of stem cell transfer, and we've come a good distance from that period of time. Don Thomas did that at the Mary Imogene Bassett Hospital in Cooperstown, New York, working for Dr. Farabee, who had recruited him there, both Farabee and Thomas having been at the Brigham Women's Hospital in Boston. Fast forward to 1990, Don Thomas and Joseph Murray received the Nobel Prize for their contributions to the understanding of transplantation. Dr. Thomas for his groundbreaking work in establishing the feasibility and success of stem cell therapy, Dr. Murray for his contributions in renal transplantation. Interestingly, both Murray and Thomas had a Brigham and Women's background as well, so one might argue that Brigham and Women's has a foundational position in the field of transplantation. On December 8th, 1990, December 8th is today, you'll recall, Dr. Thomas gave his Nobel Prize lecture in receipt of his Nobel Prize, and I would commend that to all of you who are interested in stem cell therapy, because he does review how he got to the 1990 event and the contributions he made. Fast forward 32 years, boy, have we made a lot of progress. And what, among the things we'd like to talk about this morning are the contributions and the future of, I guess broadly speaking, regenerative medicine, but also emphasizing issues of access and the challenges that we have yet to overcome, not to mention the challenges scientifically. I'm delighted to be joined this morning, and I'll introduce each of our panelists because they're going to do the hard work, To my immediate left is Dr. Stephen Grubb from Children's Hospital in Philadelphia, Dr. Linda Malkus, Dean at the City of Hope and a drug discovery scientist, as you'll hear on her own right, Dr. Stefan Thuria from University of California, Irvine, and joining us electronically is Dr. Robert Wynn, Director of the Massey Cancer Center at Virginia Commonwealth University. So thank you all for being here, and without further ado, I'll turn to Dr. Grubb and ask him to tell us what's happened since 1990 in the field of cellular therapy. Stephen, one of the pioneers in cellular therapy. Stephen?
Well, thanks for that. And, I think to just continue on your theme of cell therapy, and what we're going to talk about, I think, to a certain extent, is engineered cell therapy, which is, genetic modification of cells. But you start with the stem cell transplant experience. So we go from Don Thomas starting this off to almost, I think, over actually now 1 million people having received the transplant worldwide. And so that is the original cell therapy. And all of us, I run the bone marrow transplant program at CHOP, and obviously we're all big fans of transplant as a modality. But the fundamental thing that you're doing in a transplant, obviously, is you're doing a transplant. You're taking cells from another person and giving it to a second individual. There are risks associated with that. And one of the dreams of a lot of us in the field is there a way to do this with the patient's own cells and avoid the risks of cell transfer, which can, in some fraction of patients, be fatal, and in the beginning was fatal most of the time. And now it's not like that. The risks of fatality are low, but they're not zero, and we'd like to avoid those risks. So fast forward to 1990, at that point, they're just beginning this notion that you can genetically engineer T cells, and we talked about T cells yesterday and NK cells and cells of the immune system that are capable of actually maybe destroying cancer if you just force them to do that. And so the beginnings of the CAR T revolution are really in the late 80s, early 90s. And what's interesting about that is that nothing really happens that looks all that great for 20 years. We had to learn how to make the cells right. We had to learn how to keep the cells happy while we're doing the manufacturing process outside the body. And those are all things that were learned in that subsequent time frame. But then all of a sudden around 2010, so 20 years later, all of a sudden, you talk about breakthroughs, this was a breakthrough. So we went from nothing works, and by the way, and no toxicity because it wasn't working, to, wow, what just happened? And certainly our experience at Penn and CHOP mirrors the experience of other places, but starting with Penn, treating the first 3 patients with the adult disease CLL, something I've never seen, seeing 2 of them go into remission that lasted for many years, and then our first experience with kids, including a patient, Emily Whitehead, who I'm allowed to say it's not a HIPAA violation because if you Google CAR T, it's kind of the first thing that comes up. And by the way, Sid Mukherjee, who wrote a great book called A Song of the Cell, which just came out, wrote a terrific article about the development of cell therapy in the New Yorker a couple of years ago. And if you Google that New Yorker cell therapy, you'll see it. And it's a terrific article. But so we got to a point where we could actually do this. We could see using cells made in at a university that you could see patients go into remission. That first patient that we treated had significant toxicity that lasted about a week after her cell therapy. We learned how to deal with that. It's a harrowing story for the family and a harrowing story for the group at CHOP who actually treated her, including myself. But it worked, and then what we learned from that applied to the 15,000 or so people who've gotten CAR T since then. So there's definitely a learning curve on this, but we got to the point where we were seeing very significant, like 90% type responses in ALL and seeing in acute lymphocytic leukemia, and seeing those about half those patients, not all of them, but about half those patients staying in remission for a long time. And so that's really where the whole idea of engineered cell therapy really gets a boost. So you're taking T cells from the patient, it's a big deal. You're genetically engineering them with a virus, putting in a new gene, forcing the T cells to go after the cancer. And so that looks great, but it's all, by the way, and I think you guys know this, it's all in blood cancer, leukemia, lymphoma, myeloma, where we're seeing really great results, differing from disease to disease, but not happening in solid tumors yet. We're not there yet. So we still need that subsequent breakthrough. So that's the T cell world. And now that's cancer, and of course we're talking about the Nixon Cancer Act, but like NASA, there's stuff that comes out of that that then applies to non-cancer treatments. And so the final comment I'll make is about, back to the whole stem cell transplant issue, is we love to turn transplant into something where you're not getting cells from another person. We're not there yet. But what's odd, and this is not intuitive at all, but for pediatric transplant centers, not adult transplant centers, half the transplants we do now are for non-cancer indications to fix problems, for instance, like sickle cell disease. You can cure sickle cell disease with a transplant. There are risks associated with it. Happens, for a small minority of patients. Getting a donor is a challenge. But you can imagine that you'd like to extend that cure. And what's happening now is taking not T cells, but stem cells from the bone marrow, as many of you know, genetically engineering those using similar or quite different techniques, and then giving those cells back to the patient, their own fixed cells. And that has the potential to be curative. There are patients who are years out from these sorts of treatments who seem to have stable resolution of their blood disorder. And we're very excited about that for blood diseases like thalassemia or sickle cell disease, and there are going to be 20 more of these coming along. And so I think in the stem cell and regenerative medicine world, that's the next step forward. There's been a lot of discussion about regenerative medicine outside of engineered cells, and I think that's still way too early. We'll see where that goes. Maybe we're at the beginning of that process. But the final point is, we went from treating Emily Whitehead in 2012 to the first CAR T-cell FDA approval in 2017. So that part went pretty fast. But you have to remember the 20 years that went before that.
That's a terrific summary, Stephen, and you raise many of the questions we will expand on as we go. But Linda, Stephen summarized the process of investigator-initiated science in the cellular world. Maybe you could talk about your work in the small molecule world, because it all relates.
Thank you. So I think it was the year 2000, Time magazine had a cover with Gleevec. It was like, I don't know if they said the magic bullet. We talked about magic bullet last night, but it was like huge, and it really highlighted the transition. Dan Von Hoff is a friend and a mentor to me, and he said, Linda, when I was first starting out treating patients, and we were trying to find things that would treat patients, he said, they were sending me things like plant extracts, to look at. But wow, have we come so far now. We are able to start really interrogate the differences between malignant and non-malignant cells, and that's giving rise to a whole host of new thinking and actually new therapeutic approaches targeting the phenotype of cancer versus a non-malignant cell. So, I do a lot of NCI study section work, and I've watched the growth of, targeted therapy as they talk about it. Probably sometimes half the grants that are coming through a particular study section are really based on finding these new molecular targets and then trying to exploit them for the treatment of cancer. So my own journey, and I have to thank the NCI for this. When I was in the last century, the last millennium, when I started my research program, I had put forth an idea to the NCI into a study section, and I said, I think DNA replication is altered in cancer cells. And it didn't fly too well in the study section, but the NCI said, we are really interested in your idea, and they actually awarded me a director's award, and it launched my career. But with those resources, I was able to interrogate it. It started in breast cancer and then went full course into neuroblastoma, children's cancer, really looking at the change in the active DNA replication in malignant and non-malignant cells. And yeah, things were different in cancer cells. Cancer cells corrupt the active DNA synthesis. From there, we discovered a target, and I actually moved to City of Hope 10 years ago because of their a really unique mindset on the part of the institution to really drive translational science and their commitment to it. So in 10 years, I went from showing up with, I've got a brand new molecular target that even drug companies told me that because it didn't have an enzymatic activity, that it was actually, it had a protein-protein function, that it worked in protein-protein interactions, that it was undruggable. 10 years later, I'm very happy to say that this past September, we actually went to a phase 1 clinical trial with a brand new drug against a brand new molecular target. And this, my story just highlights what is going on in the cancer therapeutic space. I mean, it's incredible technologies on the cellular fronts, finding precision medicine, identifying patients now, what's unique to a patient. I mean, we used to, 20 years ago talk about, maybe someday, we'll be able to, a patient comes in, as opposed to putting them on a standard protocol. Everybody, if you got breast cancer, you go on this particular protocol. Maybe it's not going to be that way anymore, it's going to be subtype, subtype, subtype. And, leukemia space really shows that. So I just wanted to give a general sense that there is an incredible amount of excitement around the better defining of what is malignancy versus normal or non-malignant, and that these differences are now being heavily exploited for new therapeutic approaches, small molecules, cellular, biologic. It is a new day.
Thanks, Linda. And Stefan Surya is a cellular therapy expert. The stage has been set, Stefan, of what the directions and the progress we've been made. Maybe you could comment on your work and also some of the challenges in the future.
Thank you so much. I am actually indeed a stem cell transplant and cell therapy physician. I moved from MD Anderson Cancer Center about 3 years ago to UCI. And my goal is not only to build the transplant program here, but to continue my research in NK cell therapy. And we're very excited about this field. Many believe this may be the next important cell therapy that may be approved. Our work began more than 10 years ago at MD Anderson. I was there for about 12 years. And we started to expand NK cells. We realized that everyone has NK cells in their blood. It's one of the main anti-tumor, anti-cancer cells that will fight modified cancerous cells that occur constantly in our bloodstream. And we started to expand this because we realized that the number of cells we have, NK cells we have, or we can extract from one's blood is too small. So to obtain a therapeutic effect, and the effector to target ratio, which is probably too skewed towards tumor cell. Tumors are characterized by incredible proliferation of cells and an extraordinary expansion in one's organism. So we focused on expanding these NK cells to potentially target tumors. So we put them after transplant. And I can add a little bit to what Dr. Trump was mentioning about stem cell transplantation, which I'm very passionate about. And stem cell transplantation took off, in fact, when Dr. Donald Thomas published a paper in Blood in the 90s, I believe, not sure if it's, I don't know exactly, I don't remember the date, but it took off because he treated 100 patients with leukemia, and about 10% of them survived. Probably if we would not have seen 10% survival, I'm not sure the field would have taken off, and we have gotten from stem cell transplantation to identifying cells in the blood with therapeutic benefit. And of course, the next major breakthrough was T-cell therapy. Now, you've probably heard yesterday in the news TIL therapy from Dr. Patrick, who from Moffitt, that may work in melanoma. But continuing the same line, we focused on a different cell in the blood, which is NK cells. And we were very excited to see that we were doing stem cell transplants, and we intuitively put these cells after transplant because we wanted to prevent disease coming back. And after transplant, which in their rate of relapse depends a lot on what we treat, what disease we treat. And there are diseases, for example, high-risk acute myeloid leukemia, relapse rate would be in excess of 50%. So we said we really need to make improvement. There are types of leukemia that we transplant, but the relapse rate can be even higher than that. So we've administered these NK cells after transplant. We found extremely low relapse rate. We treated 24 patients in a paper that we published, and we saw extremely low relapse rate of 4%. And actually, one patient relapsed who ended up not being eligible for the study because had malignant cells in the blood but not in the bone marrow. So it was somewhat missed that he had this patient had 6% blast in blood and we ended up being treated, ended up being counted. But in fact, the relapse rate in eligible patients would have been 0. So this was, I think, was extremely exciting because we might be able to significantly enhance the power of hematopoietic stem cell transplantation. And this made us think that maybe we can apply NK cells outside transplant in patients with very advanced leukemias that have very dismal prognosis in general. And we've looked at a group of, in a phase 1 trial of, we treated about 12 to 15 patients with refractory acute myeloid leukemia, refractory to median 5 lines of prior therapy, and we had a 50% response rate, complete response rate in this group, and we're trying to publish these results. And we were trying to expand this. A fraction of these patients were able to get to transplant because you don't get to a transplant unless you have disease control. So taking a fraction of these patients to transplant in itself was a big deal in our minds. So I'm really excited about continuing this work at UCI. We're trying to, I put the transplant program together first, and I'm trying to get to the next step, which is NK cell therapy. Hopefully, we'll expand to other tumors where we, as compared to T cells, and Dr. Grubb really pointed out that the CAR T cells worked extremely well in these B cell malignancies. However, it seems like our ability to expand has kind of plateaued. We really need to find the next breakthrough to be able to move to a different type of cancer, different setting, and I'm hoping that with NK cells, which have natural anti-tumor properties, we could potentially find a niche, at least, for this group of for NK cells, and which will give us the opportunity to make a difference, at least in one area of cancer therapy.
Well, that's terrific. Thanks, Stefan. And you've heard from my colleagues the scientific challenges and hurdles that have been overcome and are being addressed. Dr. Nguyen is a nationally recognized, internationally recognized authority in access to therapy. You've got to have good therapy to have access to, but in addition, there are other issues that we must figure out how to overcome, not only in stem cell and cellular therapy, but in even more routine therapy. Dr. Nguyen, could you comment on this issue of access and how we get this fantastic technology into more and more patients with cancer?
First of all, thank you all for having me as part of this panel. I always enjoy being at this conference. I learn a lot, and it's just a fantastic conference. I want to apologize for my absence there. I would like to just let you know that Donald McEachin, who is a U.S. congressman, unfortunately succumbed to GI cancer, colorectal cancer, and I was at his funeral. And so I apologize for not being there in person, but it got me even more motivated about access issues. The interesting thing is when I think about access issues, I think about technology. And in fact, I've been going back through the literature and looking and with a fine-tooth comb and figuring out that, interestingly enough, with almost every new technology or every new screening intervention or medical intervention, we inadvertently increase disparities. It's a wonderful concept about what I think our challenges of the 21st century is bringing both the discovery phases and the implementation and access phases together in a way so that as we have these wonderful new therapies, we don't do what we've done in the past. An example of that is breast cancer. For many of you who are historians, you can recognize and see a trend, particularly with the SEER data of the introduction of the mammographies and the inadvertent introduction of disparities between Black and white women. The most interesting part of this for me and why this discussion is meaningful is because as we even look in the context of who are on clinical trials, particularly with CAR T therapy, we see a disparity. In fact, back in 2016, 2017, there was a number of articles written about the immunotherapy divide, which was real. The nice part about this story is that, like any other big science question, the issue is if that's the struggle, then how do we fix it or how do we deal with it? I think we're finally starting to understand that there is some aspects to certainly the bench to bedside model that has served us well. But one of the aspects of that is actually finishing that from the bedside into the community. And I think that what we've learned getting through this COVID period and others is that in addition to what we do at the bench, what we do in the context of community education, getting beyond this concept of community outreach to real community engagement matters. In fact, I recently, the AAMC just posted their 10 principles of trustworthiness, academic or institutional trustworthiness. And one of the first ones is that the community does actually have education and does have knowledge. In some cases, that's why they don't trust us. I think that we are going to have to work a little bit about understanding that it's not so much that many of these communities don't want to participate. It's just that we have not followed the principles of, one of our brilliant thought leaders of W.B. Yeats, who said, think like the wisest of men and women, but communicate in the language of the people. Frequently, we have gotten ahead of ourselves, which is fantastic to have the amazing science. But then the question becomes, for example, if minority populations are doing less well, how do we then communicate in the language of the people effectively? What I've been recognizing as a cancer center director is that I am as enthusiastic of what I'm doing at the bench and the science that evolves at the bench. I'm equally enthusiastic of the science that's now coming out of how do we better communicate, how do we better engage, and I dare say, how do we better integrate our community so that we're bringing the science closer to them so that they will actually have access. I think I'll end with sort of saying that some of the access issues, certainly, we always sort of say to folk that minorities don't want to be on clinical trials. It turns out that there's now, that's old literature. New literature is suggesting that that's actually not entirely true. when communicated effectively, many of minority populations and communities want to be part of a trial. But you do actually have to get over some elements of being able to effectively communicate. There is a level of education that's not only part and parcel with minority communities, but also rural communities as well, which we don't talk much about. Having said all that, I am enthusiastic and optimistic that after these last several years, we are getting much more of a sense that we actually have to do great at the bench, but we also have to do even better in the context of communicating effectively in our communities. And so many of my efforts here at Massey has been able to bridge the discovery and the implementation sciences and the community in a way that Harold P. Freeman and others did this way back in the 90s. This is really, I think we're on the age the age of a 2.0 version of that ability to get people to these cutting edge, high impact, and meaningful therapies.
Thanks very much, Dr. Nguyen. And you remind me of a conversation I had over dinner last night with Ms. Patricia Wiley, one of the founders of the patient advocacy movement in this country, that in order to have individuals participate on trials, you first have to be partnered with individuals in the community. I think this was a concept slow in evolving in many areas in not only oncology but other areas of medicine, but I'm pleased to say that I think there is real progress being made, but we got a ways to go. In that regard of ways to go, while we don't want to focus inappropriately on cost, Dr. Grupp has thought a lot about how you implement these wonderful therapies, but also particularly the economic barriers. Could you talk a little bit about that, Dr. Grupp?
Yeah, and there's always this natural tension between having the appropriate resources, especially to our pharma colleagues, to develop new medicines, and what that costs the healthcare system. And and I'm no healthcare economist, but I am certainly aware of the fact that we spend somewhere around 17 to 19% of our national income on healthcare. So that's a lot of money. And so the access issue, I mean, from a doc's point of view, we talk about diversity, equity, and inclusion, but that translates to diversity, equity, and access for us. And so I'll take a couple pieces of that. One is, going from clinical trials to an FDA-approved product does improve the potential for access. But in the cell therapy world, what we do is complicated, has a lot of regulatory overlay. You're collecting cells from a patient, sending them off, getting them back. It's kind of a big deal. And right now, that's really only available in large medical centers. So that's an access barrier. And people are trying to make this a little bit easier and a little more accessible, but it's really not going out to the community quite yet, and it may be a little while before that happens. Problem one. So just as an example, at CHOP, 85% of our patients come from outside our catchment area across the whole country and to before the pandemic, the world. And what we do is we have sources of money to at least get the patient there with their caregiver, put them up for the time that they need to be in Philadelphia. That's something, and it really helps a pretty good fraction of people for whom they couldn't come without that support. But if you got a job where you can't be away for the afternoon, you can't be in Philadelphia for 6 weeks. So that's an area of access that I worry about. And that's something we're trying to address. But then, yes, let me touch on the price issue. I mean, we can't get around the fact that these are extraordinary prices. So in 2017, when Kymriah was FDA approved, a clinical trial, which I was the head of the study steering committee for Novartis for, that was approved at $475,000, which at that time was the most expensive single cost therapeutic available in the US formulary. And I'm like, great. And you can argue that price. It's very complicated and expensive to make these cells. We want to make them cheaper. All those things are true, but there it was, $500,000. And the reality is that looks quaint now. So the Bluebird product for thalassemia, which is a blood disorder that's related in a general way to sickle cell disease, and we're going to see the sickle cell products come out next year. That's FDA approved. $2.8 million, and then we've just heard about an infused gene therapy. Doesn't involve a cell, but some of the similar technology for a hemophilia, a bleeding disorder, doesn't involve a cell, $3.5 million. That's the top right now, and I don't know where that goes from there. And the issue is if you have commercial insurance and these are relatively rare disorders, you get them approved. But what happens when this stuff does start working in solid tumors, as Stefan mentioned, and we're talking about much larger incidence populations, and sickle cell, again, non-cancer, 100,000 or more patients with sickle cell in the United States. What do we do about access under those circumstances? And then this gets back to Dr. Wynn's comment, and I'll stop with this, about the community. So for the last 10 years, I've been really focused on how do I get people to a large medical center and support them in that journey and get them back to their doctors where they're actually treated every day. And I think that's an important thing to think about. But now in Philly, we got a lot of sickle cell patients who could potentially benefit from this. And how do we reach out to the community to actually get them engaged, have the information they need to understand what's going on, and then get them access just to the folks in Philadelphia who might potentially benefit from this. And so that aspect of cost, especially when you get away from commercial insurance and more toward Medicaid, is, I think, a really concerning barrier. And that's where, I'm not going to argue what it costs to be on Gleevec, the drug for CML, for 20 years. I'm not going to argue what it costs to be on some of the newer targeted therapies for a year. It comes out to about the same kind of money. They're all very expensive, but fundamentally, especially when it's a one-time hit, when you're not talking about $2 million over 10 years, but you're talking about $2 million on Thursday, then that's a challenge that we are going to reach in larger incidence populations and in patients who aren't as well insured. And look to us over the next year or two and see what we can do in working with the Bluebirds and the Vertexes of the world to try to address these issues, and hold us accountable if we're not doing the right thing.
Stefan, did you want to say something?
Yeah, I have two comments actually, and I must say that practicing medicine, I realize how the barriers are at the insurance level. So I find often that there are different levels. I'm not familiar with very well with the system, but it turns out that if you have a better insurance, or if you're a better insured for a particular insurance, you have access, for example, to clinical trials. So if you do not have access to a clinical trial of this nature, let's say. That's one consideration. The second consideration, I think it's truly ironic that these therapies that are now the most expensive therapies on the planet are geared towards most disadvantaged and most socially disadvantaged and economically disadvantaged individuals. So I think this is going to be, this is probably this is overall the most challenging situation, because third-party payers are are on one side try to limit their their expenses, and how we are going to have these newer drugs accessible to this population that is where it is needed, in fact. So it's very difficult to picture, for me at least right now, how this is going to pan out with these extraordinarily high costs. And one last thing, if I want to comment on, is because I reflected after we had a brief chat before on the actual cost, and apparently the cost of a therapy is not in any way run by any government agency. So there is, it seems to me that these costs are set by the companies alone. So there is absolutely, I don't know, maybe I'm wrong, there's absolutely
You're not.
There's absolutely no oversight. So there is not a panel, a government agency that says, let's review how you came up with these costs for these therapies. So,
well, you touch on an important concept particularly important perhaps for in the Nixon Library, the library of a very skilled and internationally known politician. You're touching on some very politically sensitive areas that we're going to have to solve. But Dr. Grupp mentioned the drug Gleevec. We think of the terrific expense of cellular therapies and bone marrow transplantation. We forget that economics played into the early development of drugs like Gleevec, small molecules as well. The development of Gleevec for chronic myelogenous leukemia was slowed by Dr. Brian Drucker's challenges, Dr. Drucker, the discoverer of Gleevec, Brian's challenges in getting drug companies interested in developing a small molecule drug that would be expensive to develop and only available for 4 or 5 or 6,000 patients a year, a very small market, and many drug companies passed on that opportunity. Drug companies obviously have come around, but Linda, have you thought about this issue and small molecules?
They're coming around slowly. They still are waiting for the next platinum, or something that you can give everybody, and it's not going that way. Especially going with the precision medicine pieces now. Where you might very well be some tiny subset of somebody with leukemia or a tiny subset of somebody with breast cancer. The whole field is going this way. It's no more dumping just one big vat of one drug. You're going to have therapy cocktails of 2 and 3 and 4 different drugs at a time. So they're going to have to start thinking of more of a bar content. The bar is now serving this particular cocktail. They're going to have to think their way through that. It's not just the big bucket of one kind of drug. They're going to have, the manufacturing is going to have to change as a whole. I feel for them. But it is, there is a paradigm shifting, but it's slow.
I have to say, can I say that that's why I'm excited?
In developing your small molecule.
Well, I didn't think about that. I was chasing a target, making a drug. But even the prices of clinical trials are so very expensive. I started my own company in order to finance the clinical trials that are needed to move the drug forward. Clearly, there are going to be a ton of pressures for the cost of therapeutics. I mean, this is where we're going.
Absolutely. One thing we absolutely don't want is limit innovation. So this is absolute, and you're the perfect example here. We really don't want to limit innovation, but on the other side, it seems like there is a huge gap to how it was mentioned before, how we actually end up giving to many people that need the new therapeutics. So this is a huge challenge, I think.
But an exciting one.
Dr. Wynn, do you have
Absolutely. This is why I'm super excited. I'm excited about the science. I'm excited about not only bringing in the populations. As you're having these discussions, I was thinking about some of the recent work that got done in Montefiore and other places that showed when, in this case, when African Americans and Latinos got access to the CAR T therapy, their outcomes were statistically, there's no difference between black-white outcomes, et cetera, et cetera. But the cool part about what we were just talking about is the thoughtfulness behind the precision medicine movement. The thoughtfulness is that, for example, for me, that a woman, and I'm really glad you said what you said, is that the days of just giving one cisplatin and everybody, that those days are probably moving further and further away. And we're moving into really this new, really innovative, cutting edge space in which we're personalizing the medicine we give. That's important. I'm going to say because, for example, the work that's been done by Melissa Davis and others, say in triple negative breast cancer, has shown us that if you're Black, that's what we call it, right, which we know is a social construct, but more of Eastern African descent, you have a much more favorable outcome from triple negative breast cancer than you would if you were Black with Western African ancestry. I actually think that we've moved. The funny part about the field is not only is the basic science moving to, I think, a higher level and being much more nuanced and much more rigid and much more focused and refined, but this issue about who we're treating and who's going to be impacted by the precision medicine we give is also evolving in the context of not only understanding one's ancestry, that's the first level, but also understanding the impact that one has of place and space on their genome, right? I've always said that how the genome, that is the ZNA, that means the zip code neighborhood of association, also plays a role into your responsiveness to therapy. It's going to be ridiculously messy, but I'm excited about the mess. Because just like immunotherapy over 120 years ago, I think you just had to have bright people continually think about how to solve the problems. And then you get that aha moment, as you sort of said, the 20-year delay, where someone else comes along and says, aha, now it works. I actually think the bridge between the DNA and the ZNA, this sort of interplay that we are, particularly in the context of precision-based medicine, is going to matter. And by the way, I think that and I'll shut up, but I do think that there's one other thing that we ought to think about that we may not be able to deal with here is the criteria that includes or excludes people we ought to probably revisit. Because I don't think we've talked about those standards, about those inclusive and exclusive criteria and how they also inadvertently exclude many people who are minority without really good validation for why. So I think we're in this new frontier, if you will, exciting about where precision medicine, person-based medicine will fit into how we're going to do this for not just some, but for all. And that's the complexity of it, but that's the excitement of it as well.
And it is particularly exciting to me to hear Dr. Nguyen commenting on his enthusiasm for dealing with a mess, because we certainly have a mess. Linda?
Actually, another point, well, we talked about costs and community. Another concern, a part of the mess, if we want to go there, is actually the education piece. I mean, we have to educate the entire medical culture now. I mean, that's, we have you have this incredible knowledge, but, most everybody's out there in the community, and now we have to educate all the medical community for these type of therapies.
That's a terrific segue into perhaps an interlude where we can open the floor up to questions from the audience to ask for clarification on any of the points that have been raised. Are there questions anyone like to ask any of our experts? Yes, sir.
Hi there. I'm Flavio Roque. I'm actually representing Dr. Drucker from OHS Unite Cancer, so I'm glad this conversation came up. And I think it's very good points that the panel presented. I mean, you're talking about treating a rare disease and then the economics of that for a pharmaceutical company. But the interesting about the Gleevec story, of which, as you know, we celebrated the 20th anniversary of the drug, is that not only did it work for CML, but then there was this other disease, gastrointestinal stromal tumors, where the same mutation played a role, and similar situation, about 6,000 patients a year, not a huge number, but revolutionized the treatment of that disease. And so I think that's the other interesting piece, that you may find mutation one disease site and have it applied elsewhere. So fantastic discussion. Thank you very much.
Other questions or comments? Identify yourself, if you would, please.
I have a question that I would like to pose to you, which you just brought up so eloquently. I believe that perhaps we should question the federal government's expenditure of welfare funds to non-citizens, and shouldn't that money be redirected back to our own citizens? In other words, let's examine where our funds are actually going. And this seems like such a much more worthwhile direction for those of us that have relatives and friends that are minorities.
So I'd like to quickly give a slight perspective on that. And I have respect where that question's coming from, and I think the issue of resource allocation is incredibly important. But, you know, I'll give you a different point of view, which is I'm a pediatrician. I'm a pediatric oncologist. And if I have a patient in front of me who has cancer and I have the ability to save their lives, that's what I'd like to do.
Thank you. Terrific issue, but complicated, and I respect very much Dr. Grove's perspective. Other questions?
Hi, my name is Patricia Wiley, and I'm a patient advocate. And I just wanted to say, I want to echo what Dr. Wynn said, that things are changing now. And there are so many other groups that we need to look at. And one of the most important things I think you need to look at is the cultural aspect of those groups and what goes on, not just the color of their skin, but what their culture is, because of a lot of it would really surprise you. So I encourage all of you who work in oncology to please have your community advocates, your patient advocates, delve into that, because that's critical.
Yeah, as an old guy who's been around for long enough to see the transition, it is remarkable to look at the degree or the lack thereof of community engagement in the 80s and early 90s with cancer centers and their communities, and how much that has changed in the last 10 to 15 years, to no small extent from the hard work of folks like Patricia, who have really made us realize how critically important it is. Other question?
We're going to take one last question.
Yeah, my name is Luhua Mahmoudou. I'm representing Cal State Fullerton. Yeah, so this is actually a very interesting discussion, and I think one factor in which we have to take into consideration is the time to the treatment of the cancer after the individual have been diagnosed with the cancer. Yeah, because we actually had a research which we were actually looking at the time for which an individual started treatment of the cancer, and what are the factors that actually influences the time the individual start their treatment for the cancer. Being able to know these factors which influences the time the individual start the treatment of the cancer after they have been diagnosed is very important in actually mitigating some of the factors when it comes to the survival of the cancer. So I think it's something we have to take much consideration. Thank you very much.
Good point. Question or comments from any of the panelists on that important point?
It was just a quick thing. And first of all, I just want to tell Patricia that I am a huge fan. So I wish I were there, because this would have been outstanding just to see you in person. But short of that, I will say that when it comes to issues like time and frequency and cadence, particularly of the trials, of getting the therapies, this is where I think the bigger issues are going to come in a context of understanding how the impact of the social determinants of health play a role. It also is going to actually be incumbent upon, I would say, that the big pharma companies and the academic universities and other health systems in figuring out how to streamline the process a little bit to actually help us not only get people on the trials, but to do it in a timely manner. So I think it's again, another complicated aspect that I have had immense joy, believe it or not, over the last couple of years trying to figure out how to get people to treatment, and then how to actually do it where we're reducing the time to activation. I will say that probably not in this setting, but there are things that we're now working with other groups around the country to figure out how we can do this better. But transportation and other issues like that seem to be of no consequence when we're talking with people with resources, sometimes are the small things that get in the way of getting many of our people, particularly that we need, on the trials, getting the therapies, and getting it in a timely manner. So I will tell you that thank you for that question, and that there are at least a handful of us around the country really putting a lot of thoughts into how to come up with a system to make it better.
It's real quick. The other aspect that I am so struck by is, so I work at Children's Hospital Philadelphia. It's a great place. But it's a typical quaternary care hospital, and getting through the system, although we try so hard to make this better, getting through, it's not just getting to the hospital, although transportation is a huge barrier, and at least that seems like something you can address. But once you're there and you walk in the door, the process of getting through the system, not only dealing with your insurer, dealing with the hospital, dealing with the 42 places that you need to go, dealing with the registration process, do you have the app? I mean, all of those things are incredible barriers to care for people who don't have the prior experience to allow them to deal with these circumstances. I was treated for lymphoma 10 years ago. You want an honest answer? My wife took care of most of this stuff. And when I saw what she went through to get my care, it was incredible, and we're somewhat plugged into the system. So that aspect of just dealing with the complex systems that we've built, which are really not built around the patient, they're built around other considerations. Dr. Nguyen, tell us how to fix this.
We probably don't have enough time, although I bet Robert could summarize it in a small length of time. But we should probably thank the organizers of the conference, a wonderful opportunity for us to get together. And particularly I want to thank my co-panelists for a stimulating discussion and excellent questions. So thank you all for your attention, and it was a great pleasure for us.
Thank you.
Thank you.
Ladies and gentlemen. Ladies and gentlemen, I invite you to take your seats as we get started with our second program. Understanding the interactions between cancer and the immune system can lead and is leading to some of the most concrete and exciting developments, particularly regarding targeted therapies. To lead a discussion to expand on this, please welcome Dr. Anna Barker, the Chief Strategy Officer at the Ellison Institute for Transformative Medicine of the University of Southern California. Dr. Barker served as Principal Deputy Director of the National Cancer Institute and Deputy Director for Strategic Scientific Initiatives, and Dr. Barker will introduce our distinguished program participants. Thank you.
Thank you, Jim. Good morning, everybody. Good morning. It's a great crowd. We're so glad you all came out to talk with us today. And so think about your questions as we get started. I actually am an immunologist by training, but I'm a polymath by choice. And so what does that mean? It means that all the things you heard last night, I really believe that, at least in oncology and probably all across biological science that it relates to disease, we really need to bring our friends from engineering and physics and mathematics in, because we're at that point, aren't we? That's where it all is going, and so we have to, the elephant in the room for biology is always we didn't like math, so have to be careful with that. So I wanted to set this panel up a little bit to tell you what we hope to do with you today. First of all, we have been studying the parts of cancer for a very long time. Now, what do I mean by that? It means that, and I had something to do with this, Dr. von Eschenbach and I were at the National Cancer Institute together until he chose to go to FDA and regulate everything that I did after that. So, you know, the NCI is a regulated entity, as you might know, so we do a lot of clinical trials there. But we started a lot of activities to set the stage for precision medicine when Andy and I were together, including working together with Francis Collins to set up the Cancer Genome Atlas, National Proteomics Program, the one you heard about last night, the National Nanotechnology Alliance, and the list goes on. But one of the things we did was to set up something called the Physical Sciences Oncology Centers to do just that, to bring all these disciplines together. And I think it's going to bear heavily on what we're going to talk about today. So immunotherapy, we could talk about a lot of things today, but we're going to talk mostly about immunotherapy because it's on everybody's mind. You're hearing a lot about it. You hear there are miracle cures, and we want to make sure that we get you excited about the right side of immunotherapy, and you need to know a lot about immunotherapy and understand it, I think, from top to bottom to be able to, what I'd like to make everybody in this room an advocate for immunotherapy. I think if we do our job well today, I think you'll see why. So this is a very old idea, goes back to Cooley in the 1890s, I think, when he made a toxin that was actually out of streptococcus and a benign organism, and he figured out that he had to actually heat up the streptococcus, and he made it into a toxin to treat cancer, and sure enough, he had some responses, and so that was the beginning of immunotherapy, although some would say it goes back to the Greeks. It's a very long in the tooth idea, but one that we believe has reached an inflection point. So when I said we started by studying the parts of cancer, that's what we've done. We're in either the genomics period or the post-genomics period, but I think we're in the middle of the beginning of the multi-omics period. So right now we're measuring patients for just about everything in terms of their omics, their genome, their transcriptome, their proteome, and their epigenome are all now possible to get on a chip. So that's become the basis for precision oncology. I will tell you that about 8% of people in this country who are eligible for precision oncology are getting that kind of treatment. So if you think we don't have problems in this country with who needs to be treated versus who's being treated, you're hearing a lot of conversation about that, and you're going to hear a lot more. So let me say one more thing about immunotherapy. Immunotherapy is not new, but the concept actually in terms of oncology is quite new. We're not treating a specific target when we treat the immune system. We're actually rebalancing or redirecting the immune system. So it's a systems approach. And I believe, as you heard last night, that cancer is a complex adaptive system. And if you put cancer in the same space as you have the immune system, now you have an extraordinarily complex system. And I would predict that in 10 years, that will be absolutely the only way we think about cancer. So everything that you know about what goes on normally in your body is still going on. Cancer doesn't invent anything new. It actually takes over processes that your normal system does every single day for you. But cancer, as you know, is a very wily creature and can take over those processes quite effectively. The last thing I'll say is I think of cancer as actually a disease of systems, but also a disease of dysregulated communication. So your cells at every level is having conversations all the time and making decisions, all the time. And it goes without saying that once you dysregulate at the digital level, that's your genome, and that gets translated into changes in the analog information, that's the phenotype, that's you, you're going to have a lot of problems. And once that becomes metastatic, once that cancer evolves to the point where it can outwit all those wonderful defenses you have from your immune system, then we haven't done very well with metastatic disease in all the years we've been working on cancer. So we have a great panel today. I couldn't have chosen this panel any better because so if you look to my left here, you're going to see somebody who actually knows a lot about the basic science of immunotherapy and the immune system. And actually, I'm going to mispronounce Tom's name. I'm sure everybody does, but I'll do it too. So Tom Jaweski is the AbbVie Foundation Professor in Cancer Immunotherapy at the University of Chicago Comprehensive Cancer Center, one of our better cancer centers. Next to him is Carolyn Britton, who's the Vice President of Global Development and Early Development for all the programs at Amgen. And on the end over there is Carlos Becerra, who's the Medical Director of Cell Therapy and Cancer Research at the Hoag Family Cancer Institute, who has just been there 2 months, I think, right? Yep. Now, so I want you to know that this panel, if I go down this row of 3 people, I'm showing you all of cancer research today. So basic science and applied science, development and translation, clinical, and right next to patients all the time, changing their lives. And every one of these panelists is also a physician. So they're all patient-centric, and all the work that we're going to be talking about today is patient-centric. So Andy and I were talking before I came up here, and I said, you know, it's going to be interesting to think about immunotherapy relative to the last panel, because what we want to do actually here is to take this to a different level in terms of not just, you know, transfer of cells, some of them educated, some of them not, but how does that actually apply in cancer? What are the problems, what are the opportunities, and what do we do about it? So with that, I'm going to ask everybody to give me a couple of minutes on, here's the question. So this is a very old idea that's new again. So are you optimistic, and do you think that this might be the answer to cancer that we've all been looking for all these decades? 2 minutes. Just want to see what you think.
I guess since I'm first in line, I'll start. It's a real pleasure to be here. The point that you made towards the end, that cancer cells are not living alone, they're living at an interface with the host on multiple levels, including the host immune response, is really critical. It's been known for a long time that T cells of the immune system were capable of recognizing and destroying cancer cells. And in fact, in most patients, it was possible. I did a postdoctoral fellowship in Brussels with Terry Boone, one of the guys who discovered the first antigens that are expressed by cancers that can be recognized by the immune system. You could find T cells in the blood of a patient, mix them with the tumor cell line you derive from a patient, and kill the tumor cells. But somehow, in the patient, that didn't get lined up, right? So a big mystery was why. And what was found over the last 20 years or so is that in a major subset of patients, the immune system is engaged, the communication is initiated, the immune cells even come into the tumor. We call these hot tumors, right? T-cell inflamed tumors. But the T-cells are turned off again. And once the biology of that was figured out, these are called immune checkpoints. We now have drugs that are FDA approved in 20 different cancer entities where you block these checkpoints, you block the negative regulators, the immune system wakes up again and then destroys the tumor from within. This was, these were fundamental discoveries that led to the Nobel Prize being given to Jim Allison and Tasuku Hanjo in 2018. I'm friends with these guys, so I was at the Nobel party and ceremony and everything. There were some rock stars there too, and I played guitar in a band.
Jim was with us last year here. So.
Oh yeah, and so this was a transformational discovery. But it turns out there's a lot more to be learned. Oh, and by the way, we're talking about cure. This word came up several times last night. With patients with metastatic cancer, immunotherapy is what's actually curing a lot of patients. I treat melanoma patients. That's where a lot of this got started. And patients with metastatic disease can actually be cured with these immunotherapies. So that's the glass half full. I think that's the argument you wanted to hear from us. So I'll start there. We can come to the glass half empty where there's room to discover, I think, with a subsequent question.
Our first job here is to have you join us in really becoming enthusiastic, totally all in for immunotherapy. All right, Carolyn.
So I'm an oncologist. That means my default mode is optimism. And I am optimistic about immunotherapy. I came a little late to that realization. I started out in signal transduction. I'm another one of Dan Von Hoff's mentees, and had great mentorship from Dan Von Hoff, Gail Eckert, Dennis Slayman, mostly in signal transduction. I remember having arguments with Tony Rivas at Los Angeles and kind of at UCLA, and kind of teasing him about immunotherapy. But, you know, you have to not only be optimistic, you also have to be adaptable. And we've seen some really great responses in immunotherapy, not only in hematologic malignancies, also solid tumors. Folks have been talking about co-opting the T-cell system, especially in hematologic malignancies, but we know that that also works in solid tumors. Solid tumors are behind hematologic malignancies, but just this year, we presented data with one of our compounds in small cell lung cancer. You've heard about re-engineering T cells. One of the things that we're doing is taking a T cell and linking it to the cancer cell through something we call a T-cell engager or a BiTE. And we've seen successes not only in hematologic malignancies, but as I mentioned, solid tumors, prostate cancer, as well as small cell lung cancer. So yes, optimistic.
Go ahead.
Short answer is I'm tremendously optimistic. I've seen it. I've seen the results of a lot of the work that has been going on for many decades by basic scientists in the clinic. I'm an early adopter because I do clinical trials with all these different type of approaches. Our previous panelist talked about that we still have a lot of work to do in solid tumors, that hasn't panned out yet. I have to say I disagree with that. I've seen it myself. You're going to see some data coming up, for example, in stomach cancer, refractory to everything else, having some wonderful results with modified T cells. Pancreas cancer, pancreas cancer, yes. We've seen results. We've seen very excellent responses with several different products. So we're going to get there. It's a matter of tweaking. Give us a few years and a lot of money, and we'll take care of it.
All right. So, so part of what we're doing today, we have a lot of patients and patient advocates that are actually attending online. I know because I I sent this conference out to a lot of people that I know in the advocacy community. And it's, you know, I think it's really important that we bring this back and educate a little bit so everybody's on the same page. So, so Tom, could you say a little bit about what, you know, CAR T cells? Let's start with what CAR T means, and then how do you actually create those cells in a very brief description?
Yeah, so just to take a step back, the two main immunotherapeutic interventions that are well established now, I mentioned checkpoint blockade, where you release the brakes on the endogenous immune system. And you heard a little bit in the previous session about CAR T cells or engineered T cells, where you can take just generic T cells from the body and by genetic transduction put in a receptor that can recognize something present in that patient's cancer cell. So you're engineering it to be redirected towards the cancer type or sometimes the specific cancer in that individual. So two different approaches. Checkpoint blockade, you're reinvigorating the T cells that are already in the patient's tumor. That's maybe a little bit more straightforward. CAR T cells, you're from the peripheral blood, you collect T cells, put in the receptor of choice, grow them up in the laboratory, or now by companies. They ship that back to the center. The patient is conditioned to receive them. The product is infused. Sometimes additional therapies are given. And then the hope is that those T cells will recognize and destroy that patient's cancer. So these are two different categories of immunotherapies that are widely being applied and FDA approved in a number of different cancer contexts currently. But the technique is quite different, and what's been alluded to here is that the two fields are coming together to put them together in both hematologic malignancies and solid tumors.
Yeah, so Carolyn, talk a little bit about what are the challenges with actually making this work in terms of making CAR T cells sufficiently available in the quantities you need for clinical trials, let's say?
So I think probably there are others who can answer about CAR T cells more completely than I can. What we're working on are T cell engagers, which are different from the CAR T cells in that they're off the shelf. The CAR T cells, I think Tom's already talked about the process, right? You have to harvest the cells from the patient, send them to the drug company, bring them back, engineer them, bring them back to the patient. Patient has to undergo a lymphodepletion therapy as well. For a T-cell engager, it's off the shelf, there's no lymphodepletion, and it's available immediately. And that's one of the advantages of the T-cell engager therapy. You can use a typical manufacturing platform. I shouldn't say that because it takes away from the magic, and it really is magic in making these things. Being able to produce these proteins is complex, but we've developed systems to do it. So let's turn to how we actually administer these CAR T cells. Can you explain? You're seeing a lot of patients, right? And
yeah.
So, is there anything special about CAR T cells versus infusing a chemotherapeutic drug? Let's say. I mean, so I'm trying to get to something here. What are the barriers to actually not just creating these cells, but administering them and actually, the last panel talked to some extent about the cost of these therapies. We're not going to go into that today, but suffice it to say they're very expensive.
There is a number of barriers. One of them is what I call physical barrier, which is a distance from where people live to where those treatments are being administered, and I think that was already alluded to in the previous panel. There's also a barrier as to who is potentially eligible. For example, just one example, in stomach cancer, which is a type of cancers that I manage, with some of these modified T cells, only about 50% of the stomach cancers have the specific target, so that leaves 50% out. In addition to that, there's additional what we call exclusionary criteria that brings down the number of patients that can be effectively treated with this the smaller number, and the cost. So the cost, while the patients are on clinical trials, is is paid for by the sponsor. However, you've heard about cost of Kymriah and all these commercially available modified T cells, which is we're talking about $500,000 plus plus additional costs.
It's a lot of money.
It's a lot of money. I think the technology will evolve in doing perhaps viral transfections will change once you move to, for example, from viral transfection to transposomes and different type of approaches. The cost will go down, and hopefully more patients will have access that way. And insurance will cover.
So we want to spend a little time on CAR-T because, we spent some time in the last panel talking about the promise of stem cell therapy, and all of these adaptive cells that we actually are creating now are, I think, very promising. I think it's very promising, but we have to all sort of integrate the fact that it's not easy to make them, it's not easy to scale them, and it's certainly not going to be easy to afford them. So let's turn back to why we gave out a Nobel Prize a few years ago for the checkpoint inhibitors. And the checkpoint inhibitors have probably become the go-to therapy for lots of diseases now. And certainly in combinations. And so if you go back in the history of CAR-T, it's long. We've been at this for a very long time. Equally so for the checkpoints. So Thomas, could you say something a bit about, what are checkpoint inhibitors? Why have they been so successful, or have they?
Yeah. So in terms of the years of research and development, a lot of basic science investment was required to identify these molecules on activated T cells that regulate them, that turn them back down. The immune system is sort of designed to rev up quickly in response to a pathogen usually, and then to turn itself off to avoid chronic inflammation and autoimmunity. So it's that balance that the system is built upon, and cancer is a chronic disease, and so you end up in this negative feedback phase of the immune response, and that's what the immune checkpoints are doing. The reason checkpoint blockade is so broadly applicable to a wide range of cancers, like I said, FDA approval in around 20 cancer entities, is because that same process of immune system going on and then turning back off is relevant for many solid tumors. And so you have a large fraction of patients with many different cancers who have, you might call it a smoldering immune response against their own tumor that's now held in check by these checkpoints. And so these drugs can be broadly applicable. The one that many of you are probably familiar with, the target is called PD-1, and the two main drugs against PD-1 are made by BMS and Merck. Just to be fair, BMS is nivolumab and Merck is pembrolizumab. Again, broadly applicable in a wide range of cancers. But how do we make that better? We have patients who have complete responses that are durable. We have patients more than 10 years out from the first checkpoint blockade trials who still haven't had recurrence of their disease. And in terms of cost versus benefit, I hope we can bring up the point of if a patient is cured of their metastatic cancer, they return to society. They go back to work, they go back to school, they're paying into the tax system again. And so cure means a lot in terms of sort of feeling relevant again, and then compensating for the cost of the therapy when you are returned to the workplace. But the non-responders is what the field is really focusing on. Most patients, more than half of patients, still don't respond, and we're figuring that out, right? One of the key things that I don't want to occupy too much time here, one of the key things that we found out is that patients who have a good immune response against their tumor versus a bad immune response, one of the drivers of that is actually their gut microbiome, the composition of the bacteria in the gut, which has been shown. We were the first group to find this in the context of checkpoint blockade, and it's been extended now to CAR T-cell therapy patients as well. There are good microbes and bad microbes, if you will, that occupy our gut, that push a favorable immune response against your own cancer versus not. And if you have a good microbiota, you're more likely to respond to checkpoint blockade. This is something we can change. We've licensed some of our own work to a biotech company to create special probiotics or bacteria that could be given in different forms. People are doing fecal microbial transplantation, where, believe it or not, you take a stool sample from a responder patient and you do a transfer to a non-responder patient. It can turn them into a responder. And then diet affects the microbiota. So there are other interventions that are being developed to try and push the favorable microbes. And that's just one example where, by studying patients who fail to respond, we're figuring out mechanisms why and then trying to change that so that we can turn them into responders.
So I've raised your level of optimism, and we've, I think, supported our optimism so far pretty well. But so when I said this is a complex adaptive system, the immune system, it really is, and it talks to every other system, and I hate the word talks. It communicates with every other system you have. And so when you look at the title of this session, it's Immunology and Inflammation. Well, your inflammatory response, as you found out in COVID-19, can be a good thing or a bad thing. I mean, right? I mean, the people that ended up in the ICUs with COVID-19, they had a wonderful immune response in their lungs, but the immune response overdid it. And so those people actually ended up being very ill. So your immune system, once that homeostatic set is out of sync, it's no longer balanced. It's moving toward really tuning up the immune system. That can cause toxicities that we know can be deadly. So let's talk about that a little bit in terms of, what are these major toxicities we could anticipate in patients? And if you think about an immune system that you're trying to actually activate and you overactivate, or you activate the wrong population of cells, what have you. So what are some of the toxicities we're seeing here? What do you say, Carolyn?
So if we talk about the immune checkpoint inhibitors, they can be associated with a series of events we call IRAEs, immune-related adverse events, where you basically can get inflammation in almost any organ system. You can get inflammation of your colon, have a colitis, inflammation of your lung, have a pneumonitis. The good news is that most of the time when we're talking about these types of side effects associated with immune checkpoint blockade, it's manageable, very manageable. And the minority of patients would develop something that would be a real severe toxicity. The more common kind of IRAEs may be things that impact the endocrine system, which can be solved with, for example, replacing the hormones, things of that nature. When we talk about CAR T cells and T cell engagers, we're talking about a different kind of immune response, and that's called cytokine release syndrome. Basically, what happens is we activate the T cells, and then we cause this cascade of proteins, for lack of a better word, but of cytokines that then can cause serious issues like low blood pressure, high heart rate, shortness of breath, and some of the things you talked about when people are seriously ill from a bad infection, this sort of inflammatory response. And the good news is that in the majority of cases, serious CRS the chances of that happening is very low, and we know a lot about how to prevent it and how to treat it. So what we commonly do now is we give steroids, which kind of blunt the release of these cytokines. And we also, once the cytokines are released, we can block the effect of the cytokines with a drug called tocilizumab, for example, which is an IL-6 receptor blocker. But we're doing some really cool things to re-engineer our drugs to try to get away from CRS altogether. And some of the things that we're working on include what we call dual bites or and gated bites, in which the molecule is not only binding to one tumor-associated antigen, but it's binding to two tumor-associated antigens and the T cell. Now, why is that important? There are a minority of tumor-associated antigens that are specifically expressed by cancer cells. So some of these tumor-associated antigens could be expressed by normal cells. So if you're targeting with two tumor-associated antigens, then you're really being more specific to the tumor. So that's one approach. Another approach is...
That's a breakthrough, isn't it? I mean,
it's super cool.
Bispecific antibodies are a breakthrough.
And another approach is, well, that would be a trispecific. So a bispecific would be the T-cell engagers that are already approved that bind one tumor-associated antigen to the T-cell. A trispecific binds two tumor-associated antigens and the T-cell. So that's one approach. Another approach would be what we call masking, or somehow hiding the bite or the T-cell engager until you get to the tumor. And then once you're in the tumor microenvironment, you cleave or unmask the protein, and then it can do its job.
So isn't it interesting, Carolyn, really what you've been talking about is taking a complex system and redirecting parts of it, blocking parts of it, enhancing parts of it. Very interesting. It's an engineering. You're really a molecular engineer at this point. So are you seeing anything, Carlos, that is unexpected in patients that that really surprise you, that you thought, geez, I would never have thought that would have happened other than the miracle cures? I mean, are you seeing toxicities that you think that we can't manage or we should worry more about?
No. One of the things that I've found out over the years in dealing with these living drugs, for example, the CAR-Ts, I call living drugs, is that they're fairly predictable. So you give them, generally speaking, you give them the infusion of the living drug to the patient and, on Monday, and on Wednesday at around 10 p.m., I'll get a call.
Could you speak more into your microphone?
On Wednesday around 10 p.m., I'll get a call from the floor nurse. Oh, the patient is starting to having this and that and so on and so forth, and we have a protocol for that. So it's just a way of the cells reacting to and engaging with their target. And as Carolyn was alluding to, we already have medications to try to quell down the problem and take care of it. Therefore, the risk of severe toxicity is much, much less.
So, Carlos, when you're sitting in front of a patient and you have a portfolio of ideas, say a GI tumor. You have a portfolio of ideas that standard of care for certain kinds of cancers. How do you make a decision that an immunotherapy would be better than, say, a targeted therapy, a small molecule?
So there is a specific blocks, let me put it that way, in the clinical trial where we don't go rogue and you say, for example, somebody who has never been treated and go say, well, let's offer this to this patient yet, as long as there's some standard therapy available. But beyond that, once the patient fits a certain pattern, we discuss it with the patient and the family it's critically important to discuss it with the family too, because they're going to be supporting the patient. Once you explain it, if they're agreeing to it, we proceed.
So, for everybody on the panel, you know, we're at that inflection point where immunotherapy is starting to be certainly not routine yet. I mean, I think it's still for some tumors. I mean, and for certain checkpoint inhibitors, I think at least one checkpoint inhibitor we all know about. We seem to treat most cancers with one of these checkpoint inhibitors. There are 20 plus checkpoint inhibitors, by the way, approved by the FDA. The person who oversees oncology at the FDA often asks, how many checkpoint inhibitors do we really need, since they're targeting the same sort of antigens. But the question is, it is still a complex system after all. And you have all of these things available. So will we see a day when immunotherapy becomes monotherapy, or is it always going to be combinations? What do you think?
There are several different flavors to that question. First of all, in terms of being standard of care, I just want to maybe push back on you a little bit there, because in metastatic melanoma, for example,
yes.
Even if somebody has, if someone has a mutation in a particular gene, it's called BRAF. We have targeted therapies available and immunotherapy. The feeling was that if patients got immunotherapy first before targeted therapy, they did better, and now that's been proven in a randomized clinical trial. So immunotherapy becomes first. Now that's for metastatic disease. Then immunotherapy, checkpoint blockade, was moved to the post-surgical adjuvant setting after somebody had surgery and had high chance of recurrence. Immunotherapy now is the standard there. And now, in the past couple of years, it's been moved to what we call the neoadjuvant setting. So somebody presents with a large tumor, lymph node mass. Normally they would get surgery first. Now we get immunotherapy first, and that's been proven in a randomized trial to be better than doing surgery first.
Also in breast cancer.
Yeah, and with immunotherapy though.
Yes.
In melanoma. So now most patients would get immunotherapy before their surgery even. So it's being pushed earlier and earlier in the treatment scheme. And I just want to argue it makes logical sense. Once the cancer divides and spreads, there are more mutations that accumulate. You're more likely to get tumor cells that become resistant to the therapies that you're considering. This gets back to sort of tumor evolution that was referred to last night in the mathematical session. It makes sense to give the therapy earlier to improve the possibility of cure. Then other checkpoints. This has to do with biology. So we know that there are patients who don't respond to the current checkpoints, even though they've got a rip-roaring immune response in their tumor. By studying those T cells, we've discovered other negative regulators, and some of those are being targeted in biotech startups that I'm part of as well, right? So in terms of too many checkpoints, I think we just have to let the biology drive what we're doing and make new discoveries and see what the next level immune regulators are and so on. So biology drives.
So we've had quite a journey in immunotherapy, especially in the last few years. As you heard, melanoma was probably the first tumor beyond the hematologic malignancies to really benefit from this. And pediatric cancer patients now have been and are being treated with immunotherapy. So it's becoming quite acceptable across, I think, all types of patients. So let's talk about another problem, though, that is close to my heart, and that is how you find those patients. And so one of the issues we have is we would all like to know that we're going to go in and get access to this kind of therapy. We all have an immune system, and we aren't talking about this, but maybe we should. You should never get cancer, right? If your immune system is on target, why do we get cancer in the first place? That's a really good question, and the answer is you probably do, maybe several times in your life, for all you know. And we now have a whole group of assays coming out that say, and I think it's going to be an inflection point in cancer research, that they can find your cancer at very early stage. Now, I can't tell you that we know anything what to do, what we're going to do about it in, say, stage 1, but let's say it's stage 3. You could downstage that cancer, but more likely your immune system might just take care of that. So, we're going to be in an interesting, challenging period here, I think, for, what do you do with these assays? But let me come back to that, and let's talk a little bit about the biomarkers for this whole field. Kind of where are we? And, a small percentage of patients react, but we don't know how to find them very well yet. And so we've got biomarkers like tumor mutational burden. In other words, what's the status of mutations in a particular patient? It's not a very good measure of very much, but we also have these antigens, the checkpoint inhibitors. We can do pretty well with that, I think, Tom. So where do you guys think this is going in terms of how do we identify patients who are going to respond, and as importantly, the ones that are not going to respond? Carolyn's picking up her microphone. She has the answer.
I thought it was my turn. I feel like this is a tough question. A lot of what we're talking about in terms of CAR T cells or T cell engagers are specifically designed to work against cancers with specific tumor-associated antigens. So there, you either pick a tumor that has a high chance of expressing that tumor-associated antigen, or you can actually test the tumor for expression of the tumor-associated antigen. But not all patients that that express tumor-associated antigens are necessarily going to respond to the agent, just like any other anti-cancer therapy. Response is not guaranteed. And so this is very, very tricky. And one of the things we need to do is look at the patients who respond and who do not respond and do tumor biopsies so that we can see how do the patients differ? How do they differ at baseline, and how do they differ after treatment? And frankly, that's asking a lot of the patients who go on these clinical trials. That's asking them to undergo biopsies that are not done for their benefit, but are really done to kind of further science. And that's a huge ask. But people who have cancer in general want to see advances, and if not for themselves, then for others. And in the 20 years I was doing phase 1 clinical trials, I was often humbled by what patients were willing to do to help others.
Coming back to this concept of cancer being a complex adaptive system, and actually it's a system that's working all the time, even in the presence of cancer, maybe not as well. But what about stepping back and thinking about, what about assessing the state of the immune system in the patient as a way to come closer to identifying those patients that are going to respond? What do you think, Tom?
Yeah, so this is right where the translational research is at the moment. Usually, people used to think that metastatic cancer patients were globally immune suppressed and there was no way an immunotherapy could work. That might occur, but with solid tumor patients, it's a very small subset who get to the point of being sort of cachectic, losing weight, and being globally immune suppressed. Most patients have a still responsive immune system. We know that because we measured immune responses to COVID vaccines in all of our patients, and they made fine immune responses, right? And flu vaccines, et cetera. But the key is, do you have immune cells specific for the tumor, and where are they, and what are they doing? What's their activation status and their functional status at that moment in time? And what's making them work, and what's making them not work? And all of that can be measured with very advanced omics techniques now. And most of the action is at the level of the tumor microenvironment, so tumor biopsies are required. And you've heard about RNA-seq, or looking at gene expression in cancers. That includes gene expression of immune cells. There you can do sequencing of the mutated genes in the cancer, and some of those are connected to immune response good or bad, working or not working. And now, in the past several years, there's been a lot of work doing what's called single-cell RNA-seq. So you take a tumor biopsy, you chop it up into all the cells, and you sequence the genes in each individual cell, and you put it together like a confetti, and you see what genes are expressed by all the different cells in the tumor cells, the blood vessel cells, the immune cells. And now, this year, there's been a huge application of a new approach, which is called spatial transcriptomics, where we can take a biopsy, and in a section, we can look at all the little dots of all the different cells in the tumor and just leave them in place geographically and see who's making what. And we just have a paper that's going in where we found out something really important in responders to immunotherapy. There are two cells talking to each other at a moment in time, and if those two cells are talking to each other, those are the responders. So it means we've got to bring new cells in that have to communicate with the T cells in order to expand efficacy. So a lot of technologies are going into this, and it's the dynamic interplay between the tumor cells and the host cells that's the key to success.
I think it's really important. Carlos, you look like you're sort of disagreeing down there.
No, I follow, of course, the literature, and I can tell you one thing is that the cancer cells are very clever. They find a way to co-opt or bring in not just the immune cells, but other cells into the equation to their favor. And you mentioned about the fact that signaling, that there was a problem with signaling between the cells and so on and so forth, and that's for real. You know, the investigators have found that going back to, for example, endothelial cells, and how the cancer cells take those cells and called the endothelial cells and use them to their advantage, and take other cells and use them to their advantage, and so on and so forth. So it's quite complex, but if we all work together,
yes,
it can be solved.
So, Carolyn, some patients are actually resistant to immunotherapies right out of the gate, right?
Patients who are, all patients that I had wanted immunotherapy, but you're saying the tumor was resistant out of the gate.
That's a better characterization. That tumor is very resistant. That patient's all over it, right?
I had a lot of patients when I was in practice who wanted the Jimmy Carter drug.
I think Carolyn has a future as a stand-up comic.
When we talk about, there are a lot of cold tumors, right? So in practice, I dealt with pancreas cancer. That was one of the areas that I concentrated in. That is a notoriously immunologically cold tumor.
You want to say what that means, cold, hot?
Hot would be something that is immune responsive. Cold is something that is immune resistant. A cold tumor might have a thicker stroma, or an environment that is not permissive for T cells to enter, for example. So yes, there are some tumors that start out resistant, and we're doing, I think the field in general is working on ways to try to make cold tumors hot, and that's, has to do with turning on and off various checkpoints, or manipulating the microenvironment and different things like that.
Or were you even using viruses to make him hot, or using bacteria to make him hot? Back to Cooley's toxin from 1893. So yes, it's happening. There's a lot of different smaller companies that are in labs that are working on this.
Yeah, I agree. It's really so I have to say, just for the audience's sake, a lot of things that we don't understand, we just make up words for it. So you know that, right? So we talk about exhausted T cells. You have the feeling that these T cells are just kind of laying on lounge chairs or something. So it's kind of a strange term. But eventually, you find out that there are T cells that, for whatever, a whole bunch of good biological reasons, are not able to respond. And so, Tom, can you say more about that? Because is that I mean, is that you just, the patient would never respond, or is Carlos right that maybe you could turn that tumor on?
This is
or off?
It's the latter. This is a sort of a different terminology for the same process we've been describing using different words throughout the morning here. That when you do have, let's say, a hot tumor, you have activated immune cells that have come in to try to destroy the cancer, but they're shut down in a way. Those T cells are what have been termed exhausted T cells. I hate that term. It's actually not precise enough, right, for some of us. It seems like they're just pooped out and they just need a good night's sleep, and the next day they'll come back, but they're turned off by specific molecular mechanisms, and by reversing that you can get them to do their job again. And that's in part what the immune checkpoints are doing. And I just want to come back to this really key point that was raised, which is the cold tumors, right? The non-T cell inflamed tumors, they don't have any T cells in their tumor very often, exhausted or rested, right? Yeah. A lot of people in the field, including our own group at University of Chicago, were figuring out how to get those T cells in. It's a complex system. It's not one factor that's missing. It's a cascade of events missing in those tumors to try and re-engage or initially engage the host immune response to get those patients on the same level playing field as the hot tumor patients, so we can get these immunotherapies to work.
Before we run out of time, I want to make sure if we have questions here that you want to have a chance to ask a question. We will, if you know if your tumors are hot or cold, that would be good to start with.
I heard this morning that the studies as well as the treatments are very, very expensive. So my question is there an upper age limit to the studies or the treatments?
So for the approved checkpoint therapies, there's no upper age limit. And in the beginning, because of the toxicity risks, there can be side effects, and if they do happen, they can be they have to be managed quickly, but you worry in an older individual. I have to say that some of our best complete responders in melanoma have been 90-year-old retired people. I'm in Chicago. These, depending on whether they grew up as Cubs fans or Sox fans, they're retiring in Arizona or Florida, and then they fly up for their treatment, they get their dose, and then they go back to their retirement home or condo, their winter home, right? And they tolerate it just as well as younger people, and the efficacy is very similar.
Carlos?
Even with some of the more toxic approaches, such as CAR T, when I got involved early with some companies in designing the study, we really didn't put an age limit. I did it on purpose. And I've treated patients up to age 83, as long as they're fit, with this approach.
Interesting, too. I mean, we didn't really talk about this specifically, but for patients that are cured, essentially, of their cancer, you have memory cells. I mean, so it's a very interesting transformation that occurs in these patients. So did we have another one?
Hi, my name is Dhruv Roy. I'm from Heliogenomics. I'm glad that Anna mentioned about biomarkers for early detection. And question for the panel, how do you see this precision medicine is evolving driven by genomic and multiomics data?
Anybody want to take that?
Regarding immuno-oncology, I'm assuming, right? That's your question? Well, if it becomes once we validate it as a predictive or prognostic marker, as you can realize, it will be very useful in that setting. Early on, I think you and I talked a little bit about this early on. If we can look at the general population and find out who's at the verge of having a cancer that's going to break through, that might be another very important setting. As long as we know what to do with that information,
yes.
And if we were able to change the natural course.
So we're going to run out of time, but I want to give our panelists a minute each. This is a real challenge to tell us what is the most important thing we can do to move this field forward. And I say that because if I were running the world, I would be putting a lot more resources into this field, and we're not. And I don't know, I sit on the National Cancer Advisory Board in Washington, but I don't see it. I mean, we have a moonshot. I think we need a Mars shot for immunotherapy. That's my own personal bias, so I'll just put that out there. And if I were still in government, we'd be having, you know, one of these, not a human genome project, maybe an immunotherapy national project, but I think it really needs a shot in the arm. So start with Tom. 1 minute. What would you do?
Yeah. For me, it's understanding failure. So now we have drugs that are effective in a large fraction of patients, and we need to figure out why they're not working in the other fraction. And so we need to, and this is what we do in our program, we're banking everything for multidimensional omics, germline DNA, serum metabolites, tumor exome sequencing, tumor RNA-seq, gut microbiome sequencing, et cetera, and understanding the multidimensionality of mechanisms of resistance. So the patient now becomes the center point, and we call it reverse translational research. We figure out all the different dimensions of mechanisms of failure, like all the different ways your car can break down. There's one way to put all the parts together to make your car run. You take any one part away and it can fail. So too with immunotherapy. We're cataloging all of those. It's an AI problem in many respects. And each of those patient observations goes back into the lab for a clinically relevant basic science project and a discovery platform.
Well, Tom, just like we're finding, you know, we're finding drugs that work initially in one cancer, and then we start looking at other cancers, and obviously the pathways are very overlapping. It's a complex system, and I think, you know, I think there is an opportunity here to treat every patient, maybe in combination, but I think there's an opportunity to treat every patient with immunotherapy in some ways. So, Carolyn, what do you think?
For me, it's really finding ways to work together. I mean, we all spent 3 years, maybe 2. It seemed like 5 in our own little silos, in our, you know, on our WebExes. But we've really got to figure out ways that we can partner with academic institutions and industry and remove some of the gunk that gets in the way. And by gunk, I mean bureaucracy. And we need to find ways to partner with patients, especially, and take into consideration what are the things that are most important to them, and what do they consider is reasonable to undergo on a clinical trial, and what is not reasonable. And then I would totally agree with what Tom said, and learning from past experience and failing forward.
Carlos?
I advocate for the community here. The majority of patients are managed in the community. And again, this requires a partnership with industry, basic of labs or basic science labs. But we all have to understand that the human being is very heterogeneous, and it requires a lot of information, a lot of input from a lot of patients and different backgrounds in order to answer this question. And that information will come from patients that are managed in the community.
So put the patient at the center. Put the patient at the center and start really developing an understanding of, you know, what is the state of that patient immunologically from the time they become a patient until they're treated, and then post-treatment would be very helpful. The other issue that was raised I thought was really quite profound, and that is, in many ways, this disease is the disease of outliers. We should study the outliers a great deal more, the super responders and the non-responders, and I think we could use that database. I hope that what you take away today is that this session actually is talking about immunotherapy, but we're really talking about a change in the way we view biology and the way we view the disease we call cancer. Essentially, it's really a disease of a very complicated, complex system. And people always ask me, why are we not making progress a lot faster? Well, we have been studying the parts of cancer, and that was important. Very important to know the changes in the genome, very important to understand all that. But now we're at a point now we need to understand that from the point of the patient and the system that that patient represents, and I should say the complex adaptive system that that patient represents. At our institute, we talk a lot about coarse-grained biomarkers. That is, biomarkers that are more reflective of the state of the patient, and as somebody that works at the molecular level, we spend a lot of time down at the molecular level. How does that translate into what you see in patients, especially around the immune response? But I think we're all optimistic, and I think this has been a great discussion, and I want to thank our panel and thank our organizers, Dr. von Ischenbach and Marlene Malik and Ellen Siegel, who can't be here, and all of you for being here, and all the patients for helping us in science to understand that it is the phenotype, it is the patient that's important, and we're very aware of that, and we need more of those kinds of partnerships. Last thing I'll say is everyone up here has also a relationship to the private sector. They've either started companies, or they're in a company, or they're part of that process. We need more of that in terms of translating this information much more rapidly into patients. So thank you all for listening today.
Ladies and gentlemen, that concludes the second panel of the Nixon National Cancer Conference. We ask that everybody exits the room to allow our catering team to come in and place lunch. We'll join back here at 12 o'clock p.m.
So let's get started. We're grateful to the Hoag Family Cancer Institute for their sponsorship of the Nixon National Cancer Conference, and especially for their sponsorship of today's luncheon. The Hoag Family Cancer Institute is located in Newport Beach, California, and boasts the largest Phase 1 clinical trials program in Orange County. To introduce our keynote speaker, it is my pleasure to welcome Dr. Burton Eisenberg, the Grace E. Hoag Executive Medical Director, Endowed Chair at the Hoag Family Cancer Institute. He is responsible for strategic planning and the Hoag partnership with the USC Keck School of Medicine, and leads the development of world-class cancer programs for the Orange County community. Please welcome Dr. Eisenberg.
Thank you. It's my pleasure to represent Hoag and the Hoag Family Cancer Center during this conference. The Hoag Cancer Program is a community-based program. It's grown quite considerably over the past decade. We now see and care for well over 4,000 new cancer patients per year. And as has been noted in this morning, about 80 to 85% of cancer patients in the United States are seen and managed in community cancer centers. It's important, if you know anything about Orange County, if you're a new cancer patient or a cancer patient undergoing active therapy, the last thing you want to do is get in a car and take the 2-hour drive to LA. So having a complete cancer program within the community has really, I think, been an important additive. I'm also quite proud of the fact that we have developed and resourced a considerable effort in cancer research, combined with strategic recruitment of a cadre of physician scientists. This provides and has been providing for a symbiotic outlet for the existing and exciting translational cancer science coming out of industry. 100 days, that's a pledge that we made about a year ago and was discussed a little bit this morning about how to be efficient in opening clinical trials. One of the problems with getting clinical trials open and finding eligible patients is the process of getting the trial from industry, getting it into the clinic, and then finally getting it approved. So we've placed a quality improvement on 100 days. 100 days or less from the time that we sign the CDA to the time we get the go letter is the goal. And I think that's going to help a great deal around the country in various programs in order to enhance the clinical trial portfolios. So it's my honor to introduce today's lunch speaker, particularly because he's a fellow surgical oncologist. I just want to provide some background. So Dr. Andrew von Eschenbach, he received his Bachelor in Science and Biology from St. Joseph University and his MD from Georgetown. He also then served in the U.S. Naval Medical Corps with the rank of Lieutenant Commander from 68 to 71, and then began his career at the University of Texas MD Anderson Cancer Center in 1976, where he went through a series of progressions from urologic oncology fellow to eventually becoming chair of the Department of Urology in 1983. He was the founding director of the prostate cancer research program at the Anderson and director of the GU Cancer Center as well. In 2001, he was selected by then President George W. Bush to serve as the 12th director of the NCI. Finally, in 2006, he was actually appointed the commissioner of the FDA, which he served until 2009. And following that, Dr. von Eschenbach has been very active in industry and support groups and continued his passion for working towards better therapies, particularly in the area of prostate cancer. And it's my great pleasure to introduce Dr. von Eschenbach. Thank you.
Thank you, and I meant what Jim said. Please continue to eat and enjoy your lovely meal. It will not bother me or slow me down a bit, I can assure you. Last night, I stood here totally, completely stunned, and for some of you who know me well, were shocked to realize I was actually even speechless. But that honor and award that was bestowed upon me by the Nixon Foundation and the Nixon Library is something that I deeply treasure, and I am incredibly honored to be associated with this library and this foundation, especially as we go forward. Presidential libraries are places of repositories of history, and this is certainly one of the most beautiful ones, and I think also one of the most meaningful ones. But more importantly, this Nixon Library is not only a repository of history, it is creating history. It is taking on the opportunity and the initiative to have conferences such as this, where we look back at the history of the National Cancer Institute, but we more importantly are looking at the trajectory and the future of where that history is leading us. Last year we came together and looked at the impact that the Act had on the evolution of science and the evolution of cancer care. This year, looking more at how that progress is impacting fields of science and medicine in general, like the ones we just heard about this morning in stem cells and immunology, and later with regard to nutrition and digital science. And so we're on a trajectory, and what I want to kind of share with you in the time we have at lunch is where that trajectory may be leading us from the perspective of the convergence that I see that's occurring in both science and technology that's influencing, because of the progress in cancer, because of our ability for the first time to understand the fundamental genetic and molecular and cellular mechanisms that underlie this process that we call cancer, that we're now able to expand and apply that much more broadly across the whole variety of diseases, and where there are new tools and new opportunities that are coming in, which are going to be able to catalyze and rapidly accelerate that. So we know that cancer as a process has its initiation and susceptibility to the point where it progresses through local growth, metastasis, and ultimately causes the suffering and death that we see all around us. But we're also beginning to understand that we can now strategically intervene in that process and be able to find critical points along the way where we can intervene, maybe not just in a single way, but in multiple ways in which we can then either prevent, eliminate, or modulate and control that disease. And so you're beginning to hear new commentary in the lexicon where we've moved from seeking a cure for cancer to now being able to think about, if not curing, preventing and even controlling, modulating it so that cancer becomes a chronic disease, but one that does not cause the suffering and death that we have been so plagued by. One of the things that has helped us in beginning to understand this is the work and the progress that the National Cancer Act enabled refreshed our way of thinking about cancer. And it goes back to an old theory over 100 years ago by Paget, who said or proposed the seed and soil hypothesis, that what was occurring with regard to cancer was this dynamic interplay between the seed, the cancer cell, or the tumor, and the soil, the host that tumor cell found itself in. And what you've been hearing in this conference today, and what you'll hear more of a little bit later, is the progress of the past since the National Cancer Act has given us extraordinary new insights into the tumor, into the cancer cell. But it's also refreshed and opened our ability to begin to think about the soil. And whether it's the modification of the soil at the microenvironment or the macroenvironment, we're now beginning to see that trajectory lead us to be able to solve this problem by being able to work both sides of the equation. So we heard a little bit earlier about some of the inferences of the microenvironment. The fact that immune cells have to then interact with the environment that the cancer cell finds itself in, and we're learning how to control or modulate that microenvironment to be able to attack that tumor cell much more effectively. And you'll hear later about our ability to look at the total macro environment, the entire person, whether it's from their perspective of their immune system or their nutrition or also the gut microbiome. And so what we're seeing is this emergence of this opportunity and this trajectory that's taking us to an ability to see the tumor and to see the problem in a totally, completely new and more effective way. That's leading us to begin to think about taking that knowledge and applying it to much more disparate problems. And cancer research and the progress that the National Cancer Act made possible has really impacted, even today, diseases, for example, such as COVID. That whole idea of the seed and the soil, the idea of the interaction between the tumor and the host, has been exactly what's played out in COVID. It's a virus. It has its own unique properties. It mutates and creates its own identities, but its outcome is very, very much dictated by the host or the person it finds itself in. And so we've seen this wide expression of COVID as to whether it's just a mild respiratory tract infection like the flu, or whether it in fact places one in an intensive care unit and creates a lethality. Our ability to understand that kind of dynamic interplay from cancer will help us be able to understand that dynamic interplay between all of the other disease entities, both acute and chronic. More often, or moreover, beside that, the progress over the past 50 years has really enabled science and technology to evolve in many other ways and in many other areas. And what I want to kind of emphasize going forward is this idea of convergence, that we're beginning to see these other advances come into the opportunity to be able to transform our entire way of approaching cancer. So to be specific, I want to point out two areas in which that's occurring by virtue of the integration of the physical sciences and the life sciences. You heard a discussion last night about the role of physics and mathematics, and it is true that with the understanding of these fundamental mechanisms of cancer, biology has become a digital science. We now are able to begin to understand elements of information and data that we can now process and be able to utilize in a way of data management that's enabling us to begin to understand cancer not just simply as an event, but more importantly, as a process. One of the other really important areas of convergence for cancer in terms of the integration of the life sciences is to begin to see that there are fundamental underlying mathematical principles that govern the biological outcomes that we observe. And there are aspects of physics that are also operative in what we view as biology. There are physics principles and mathematical principles that govern, for example, the protein folding that occurs that's responsible for functions with regard to cell, and that field is opening up enormous opportunities in understanding protein misfolding diseases such as that occurs in Alzheimer's disease. There are mathematical principles underlying biology, and some of you in the room may be familiar with the term Fibonacci numbers. But way back a couple centuries ago, a mathematician came up with the idea of the series of taking numbers 1 and 2, adding them together and continuing to add the subsequent number so that you get a series. That series, if you divide the former by the latter, gives you a ratio that's constant of 1.6. I didn't intend to give you a lesson in mathematics, but that fundamental underlying sequence of numbers and that golden ratio determines and dictates the geometric formulation of a nautilus shell, the branching of a tree, the arrangement of petals in a flower, and the design of the human face. And so what we're beginning to see is that this convergence of science, math, physics, engineering are enabling us to see biology through an entirely new prism and begin to integrate and converge our knowledge of basic biological processes and our knowledge of the underlying physical laws and dimensions. The other area that's quite, I think, for me, interesting is with regard to our understanding of these the physical sciences, is the application of physical energy to biological systems. Western medicine has traditionally kind of perceived the cell as a bag of fluid in which, in an aqueous state, chemical reactions occur. And so if we want to perturb the biological processes of a cell, alter the energetics that are occurring, driving those things, we tend to do that pharmacologically with a drug. So it is a chemical model of a cell based on the fact we think of it as a bag of fluid. If you think a little bit about Eastern medicine, there tends to be a perception of the cell as a solid state, similar to what you might think of as a transistor or a microprocessor, in which the cell structure is able to then communicate with the biological functions through electromagnetic energy. And that has led us to begin to understand that when we apply physical energy to a biological system, we get a dynamic interaction, which we've used mostly for the purposes of getting diagnostic outputs. So X-rays, sound for ultrasound, magnetic fields for MRIs, we've used those to perturb that system to get a readout that gives us an image. But what we're learning more and more through this kind of research we've catalyzed is that applying that physical energy is actually perturbing the biology of that cell. The most gross example of that is, if you will, the application of ultrasound that's focused and of high energy to basically denature the protein and cook a cell. There's also being able to modulate the ultrasound in a way that instead of producing heat in the cell, it produces cavitation and essentially blows up the cell or shreds it. And that is giving us enormous opportunities to go back to a conversation that occurred earlier, which is how do you perturb the microenvironment of of an immunologically cold tumor like pancreatic cancer that's so dense, now applying focused ultrasound, disrupting the microenvironment, breaking it down so that it goes from cold to hot and allows the immune cells to now have access to what was inaccessible. This kind of concept of the physical energy to biological systems will have enormous implications in a whole variety of fields. From the perspective of the immune system, we now know that you can apply electrical energy or ultrasound energy to the vagus nerve, and that will create the release of lymphocytes from lymph nodes that are in the intestinal tract. You can also do it by applying ultrasound to the spleen, and when doing that, the spleen will reduce white blood cells or lymphocytes, and depending upon the parameters that you dial into the ultrasound, you can get different kinds of T cells or lymphocytes with different cytokine profiles. So the idea that you can begin to modulate and control the body's immune system, not with a drug, but with physical energy. This field is emerging. It will move from oncology to other areas like neurobiology, and we're already recognizing that you can use physical energy to alter the blood-brain barrier, which allows us to be able to deliver drugs for brain tumors that previously were unable to get into the brain. And there is work, as obviously has been done, to deal with issues like Parkinson's and even now depression. So the progress and the trajectory that the National Cancer Act created has enabled us to move well beyond where we are with regard to our ability to begin for the first time, treat, manage, and eliminate the threat of cancer in ways that we were never able to do before, to now be able to see that we're moving into an entirely new realm where the progress here is leading to a change in medicine as a whole. It is a new era of molecular medicine based on the progress that that National Cancer Act of 1971 catalyzed and enabled. And so the past is a prelude to the future. The National Cancer Act of 1971 lives on because of the vision and the leadership of President Nixon. Its accomplishments continue to have wide-ranging ramifications, and it's placing us in a position where we can begin to think about an opportunity to transform the future of millions and millions of lives. One of the things that occurred to me during my career was individuals who were interested in writing a book about what was it about cancer patients that seemed to make them heroic and their ability to endure and even overcome their disease. And upon reflection, it seemed to me that there were three characteristics of those patients. They had faith, they had hope, and there was love. The faith was that they had belief in the value of their life and that the fight was worth fighting. Hope was that there was an expectation that tomorrow could be better than today. And love was simply that someone else cared. They were not in this alone. We are at a place where you heard last night Mauro Ferrari refer to love as a word that needs to be imbibed in what we're doing. All of you here today are here because you care, because it makes a difference to you that others will do well. You are creating that kind of love that will enable that kind of heroicism. The National Cancer Act created the hope. The National Cancer Act enabled us to begin to see that tomorrow could be and will be better than today because of the kind of progress that you're experiencing and listening to in this conference. And finally, President Nixon and those who joined him And that National Cancer Act of 1971 created the faith and the belief that we would and will do what is necessary to change the world and be able to provide for the people and the patients who are so desperately depending upon us and you. And so it's a great privilege for me to be associated with this conference. It's a great privilege for me to have walked this journey with so many of you, and I am enormously grateful for the honor in which you've bestowed upon me. And as you create that future faith, hope, and love based on that progress, I wish you God's blessings. Thank you.
Continue to enjoy your lunch, and our next session will start at noon. Thank you.
I guess, was it Andrew? When will we know that we're live? Are you gonna flag us? Oh, he left. Okay, well. And your husband in DC, that was fun.
And you guys made it back, and likewise.
Zoom and get ready for our third program on nutrition. We've all heard and are perhaps acolytes of the great adage, "You are what you eat." Well, it wasn't too long ago that our understanding of what we eat and its effects not only on our weight, but on our very health and well-being were vastly understudied and arguably underappreciated. But within the last decade, marked advances have occurred in this field, especially related to cancer prevention. So it's now my pleasure to introduce Marlene Malik, who has been the inspiration behind the Nixon National Cancer Conference from its inception. In addition to being an active and registered nurse, she co-founded with her friend Dr. Ellen Siegel, Friends of Cancer Research, a nonprofit advocacy group based in Washington, D.C., and now serves as vice chair. In addition, she is a member of the board of directors of MD Anderson Cancer Center and Duke University's Cancer Center, and perhaps most importantly, she is a member of the board of directors of the Richard Nixon Foundation and a friend. So please welcome Marlene Malik.
Good afternoon, and following our healthy lunch, it's appropriate to move into the important topic of nutrition. So just a few months ago in September, the Biden-Harris administration hosted the White House Conference on Food, Nutrition, and Health. Now, this conference came 52 years after the first White House Conference on Nutrition that was organized by President Nixon. Yeah. And at that 1959 Nixon administration conference, 5,000 participants developed 1,800 recommendations to overhaul the country's effort to combat hunger and malnutrition. In a successful example of bipartisanship and collaboration between the public and private sectors, almost 90% of those recommendations were implemented, resulting in major expansions to the food stamp and national school lunch programs, and the creation of a supplemental feeding program for women, infants, and children. It's called WIC, and the development of dietary guidelines and nutrition facts labeling. So many of the successful initiatives from that conference are still being used to guide health and nutrition to this day. The importance of the interplay between diet and cancer cells is acknowledged and accepted. For patients, new research and development in this area of particular interest, relevance, and importance. So today's panel on nutrition is moderated by Dr. David Heber. Dr. Heber is the founding director of the Center for Human Nutrition at UCLA, where he has been on the faculty of the School of Medicine since 1978. He is Professor Emeritus of Medicine and Public Health, and he is founding chief of the Division of Clinical Nutrition in the Department of Medicine of the David Geffen School of Medicine at UCLA. He is also former chair of the Medical Nutrition Council of the American Society for Nutrition, which is the largest scientific society in nutrition. And he is also a fellow of the American Society for Nutrition, which is the highest honor the society confers. So please welcome Dr. Heber.
Thank you very much. This afternoon, we're going to have a session on nutrition and cancer. As was mentioned, I've worked in this field since the late 1970s, and I'm sure my early research benefited from the National Cancer Act signed by President Richard Nixon. Today, I'm joined on the panel by Dr. Will Lee, who's the CEO and president of the Angiogenesis Foundation, and Dr. Richard Lee, who is director of the Integrative Cancer Program at UC Irvine, which is part of their Department of Support Services. I probably got the titles quite wrong, but in any case, he's a leader in integrating cancer with supportive services, including nutrition. And then remotely, we have Dr. Daria Mosafarian, who's special advisor to the provost and dean for policy of the Tufts Nutrition School, the John Mayer School, and he's the John Mayer professor at that school. And he's this year taken a special effort to activate information to the public from the White House Conference on Hunger, Nutrition, and Health, which was mentioned. And then we also have Dr. Susan Main, who is the director of the Center for Food Safety and Nutrition at the FDA, which we call CFSAN, and she's also been an active researcher in nutrition and cancer. So I'd like to start my comments by doing something very practical. Today we had a wonderful healthy lunch that Dr. Lee and I and other members of the panel helped to design. And what you may have noticed when you looked at your plate was that there were multiple colors on the plate. There were red tomatoes, purple blueberries, green arugula, which was different than the green bok choy and some of the other lettuce. And then you had wonderful fruits, citrus fruits as well. So you had a representative of many different plant families. And I think it's important to note that humans evolved on a plant diet that had all these various varieties. And these substances are antioxidants. They also go into your body to various tissues in your body. They localize in those tissues. We have some wonderful examples of that. And they are anti-inflammatory. And maybe, Will, would you like to comment a little bit about angiogenesis and what it does there?
Well, one of the nice things about this conference is it's really based on the fundamentals of understanding cancer biology, which is a rapidly moving plane with lots of cellular, molecular, and genetic biology tied to our immune system, and then broadening it out to the microenvironment and then to the macroenvironment, and then to behavior. And behavior is really where food comes in, because we all make choices 3 times a day, sometimes more. And we're beginning now to understand how to pull all these things together. And one area that ties together cancer biology, the micro and macro environment, and our behavior is our circulation. And how our circulation grows is through a field called angiogenesis. Angio meaning blood, blood vessels. Genesis, how it grows. We all have 60,000 miles worth of healthy blood vessels in our body that bring oxygen and nutrients to every cell, every organ in our body. And how what we eat actually influences the health of those blood vessels. Cancers can hijack those and corrupt those blood vessels, so they pull vessels to feed themselves, without which a cancer could only remain small, about the head of a matchstick or smaller. But once the blood vessels touch the cancer, the tumors can explode 16,000 times in size. And the same blood vessels that feed cancers also allow cancer cells to escape into the circulation to metastasize. So our ability to keep circulation healthy, that's really our whole cardiovascular system, is in fact really tied to food. So certain foods can help to normalize, help to streamline our circulation, but also can help to cut off the blood supply, the errant blood supply that cancers might try to take. Some of the foods that are out there, edamame, which you actually had, soybeans, mushrooms, tomatoes have lycopene, which can do this, green tea, a whole series of actually foods that contain bioactives have been studied in the lab, can studied in the clinic, and also studied in population studies as well.
Yeah, and we can go with angiogenesis not only down to the level of the tumor cell, which can only grow a few microns in size without growing new blood vessels, but we do this in other parts of our body. We do this in the heart. When you exercise, you get low oxygen in certain parts of your heart. the heart grows blood vessels. So in that part, angiogenesis is very good. There's another place where you grow them. It's going to lead me to our next panel member, and that is the central stomach. You know, we talk about the freshman 40. People go to college, they eat too much, they exercise too little, and next thing you know, this thing grows like a tumor, and it grows very rapidly. And the cells outgrow their blood supply, and the cells die and send an inflammatory stimulus around the body. And this is so prevalent in America today. 1 out of 3 Americans is obese, 1 out of 2 Americans is overweight. And this is a pandemic in our country. It's epidemic around the world, but here in the United States especially. And I'd like to ask Dr. Mozafarian if he could comment on what's happened in this country between 1970 and 2010 to the standard American diet, which we call the SAD or SAD diet. And Derry, could you comment on this? I know you've studied it extensively.
Well, yeah, obesity. So first, it's a pleasure to be here virtually. I was hoping to be able to get there in person, but was not able to do so. And it's also a pleasure to celebrate Nixon's legacy, not only of the National Cancer Act, but of the 1969 White House conference that he did with Jean Mayer. You mentioned I'm the Jean Mayer professor. Jean Mayer, who founded our school, organized that conference with President Nixon. So it's really a pleasure to be with you. I'll talk about obesity, but I hope we can also later talk about the direct effects of foods on cancer, because from recent research that we've done, those effects are larger than the obesity-mediated effects. But in terms of obesity, this is not only a national epidemic, it's a global epidemic. There's not a single country in the world that has had even a plateauing of obesity. We have a global systems problem of obesity. And what's striking about it, I think, is that while clearly it's related to changes in food, especially in our country, physical activity hasn't changed that much. Obviously, our genes haven't changed that much. Food has clearly changed because of the Green Revolution and processing of food and many things that were done in the last century to create shelf-stable, sufficient calories for 8 billion people. Because of those very conscious decisions, we've had industrialization of agriculture, industrialization of food, and that's dramatically changed our biology of interacting with food in a way that's led to obesity. And it's not just led us to eat more. And I really want to emphasize that we published an analysis just a few months ago showing that while between 1970 and 2000, calorie intake steadily went up in the United States as we became more obese, after 2000, calorie intake has actually plateaued or even gone down a little bit while obesity has continued to rise. So there was kind of this initial hit of processing of food, monocropping. We can talk about why that might have led to obesity, that led to the obesity epidemic. And now we have a self-fulfilling vicious cycle of obesity where even without calories going up, we are getting more obese. And I believe this is due to much more complex biology than just calories in, calories out, related to impacts on our microbiome and how much of our calories our microbiome consumes versus our bodies consumes, effects of small non-coding RNA like microRNA that gets transmitted generation to generation, effects of diet quality in the food we eat on actually metabolic expenditure, on mitochondrial use of ATP and electrons. There's as evidence from animal experiments that the kind of food we eat actually affects energy use in the mitochondria, browning of fat, brown fat, adipocytes that give off heat and burn energy. So there's so much interesting complexity, and I think what we've learned is, again, we have a global obesity epidemic. It's definitely due to changes in our food system as the primary cause, and we don't totally understand what's happened, especially in the last 20 years, but it's clearly causing more cancer.
Yeah, it's very interesting. You brought up the microbiome. And about 10 years ago, I met a professor at UCLA named Sid Feingold, who was in his 90s and had a laboratory where they could grow the bacteria of the microbiome over at the VA hospital. And we started a collaboration working on the microbiome. And I can say the microbiome has revolutionized nutrition science. And with what happened at MD Anderson and immunotherapy that you heard about earlier this morning, it's no question that the microbiome importance in terms of therapy of cancer suddenly went way, way up. So we look at this microbiome, and of course the first idea is, oh, I'll just throw a probiotic in there, I'll find some fiber that'll change the microbiome. No, the microbiome is a forgotten organ in your body. It's 14 trillion bacteria, 1,000 different species of bacteria that live in communities and share DNA among themselves. And they make things from what you eat. So what you eat, what you don't digest and you pass down into your colon, gets fermented into other compounds, short-chain fatty acids, substances like indole propionic acid that go to your brain and act as antioxidants in the brain. So this is a systemic microbiome, just as cancer is systemic. And the really exciting part is that 70% of your immune system cells are located in the intestine proximate to the microbiome. And those bacteria interact with the immune cells that are going down there and sampling what that microbiome is like. So today, we're developing something new. There's a $170 million effort at the NIH called the Nutrition for Precision Health Initiative, and there are 6 clinical centers around the country today looking at nutrition in a new way called precision nutrition, where you look at the anthropometrics of the person, their body weight, their body composition, but their genetics, their epigenetics, the microbiome, their physical activity, their sleep patterns, their stress level, their environment, their socioeconomic status, all these things put together. Why do I bring it up? Because after the National Cancer Act in 1971, the randomized control trial was the way that cancer moved ahead. They were able to systematically study different forms of chemotherapy and other therapies to help cancer patients and move the field forward. However, when we do a randomized control trial, we enter in those subjects and we assume they're all the same. Well, we'll do some and compare. Well, they were about the same weight, their gender was, oh, they're not significantly different. But we don't talk about the detailed biology of each person. And that's the future that's very, very exciting, is to look at how we can take the cancer patient and integrate what we know about nutrition into their status and look at how that affects responses. And that's the very exciting area where we can merge precision oncology with precision nutrition. I think that's a really exciting area. And I'd like to now go to Susan Main, who also participated in the White House Conference on Hunger, Nutrition, and Health. And Susan, could you tell us briefly, I've known Susan for a long time. She's actually quite a distinguished scientist who worked on some of the early trials in beta-carotene and various types of cancer. But what I'd like you to talk about today is what is the federal government and the FDA doing about this issue of nutrition and cancer?
Great. Thanks, David, for having me here today. And yes, I was a researcher for many years working in cancer prevention and control, across the whole continuum, survivorship, all of these issues. And the science is exciting. The investments that the federal government, including NIH, are trying to make on the precision nutrition side are also very exciting. But in the meantime, we need to do what we can to turn this tide of diet-related chronic disease and the obesity-related conditions and metabolic syndrome. And that was what really motivated the White House Conference to come together. It's hunger, it's malnutrition, it's epidemic of diet-related chronic disease, and what can we do as a federal government and all of society to try to address these problems now, the urgent need to turn the tide. We know our healthcare costs are largely driven by a lot of these diet-related chronic diseases, and we need to do better. So I was honored to be one of the participants in the White House Conference this year, really highlighting some of the tools that FDA has through things that many of you may not even realize in the past. We didn't have access to the information, things like the nutrition facts label. I'll note we updated that in 2016 to put added sugars on for the first time so that consumers have additional information on added sugars. But we're also working on how to create a healthier food supply for everybody. We mentioned behavior change and the importance of behavior change. We're also looking at FDA and in the federal government is how do we create a healthier food supply for everybody in the absence of behavior change. And some of the work that we've done, some of it may more impact the cardiovascular side of chronic disease risk. But I will note that cancer survivors are at heightened risk of cardiovascular disease as well. So examples where FDA was able to take actions to essentially remove the artificially produced trans fats, the partially hydrogenated oils from the U.S. food supply. That creates a healthier food supply for all using the authorities that we have. We've been working also on sodium reduction to try to lower the amount of sodium in the U.S. food supply, which can help reduce cardiovascular disease risk. I'll also note sodium is implicated in certain cancers like gastric cancer as well. So we're working on creating a healthier food supply for all and also starting earlier in life, because we've all seen once a child is on a trajectory, starting with overweight, the likelihood that that child will grow up to be an overweight or obese individual is very, very high. And so much of our work is also focused in on early in the life period. Some examples are trying to create healthier school meals for the children in this country. And that means more fruits and vegetables. That means lower sodium. That means getting children adapt to healthier moods so they start on a healthier trajectory for the future. You spoke about the different vegetables, the phytonutrients, the colors there. We need to get those things into the diets of our children so that they get accustomed to those. They know what they are. They become familiar with them, and that becomes incorporated into their lifestyle. So we are working with USDA and in partnership to try to get healthier school meals for all. So really translating all the exciting science that you're all talking about, but really bringing it at the macro level to the populations to try to impact public health at a more, high level. And it's not just here in the U.S., it's globally, because as you heard, the obesity epidemics and some of these diet-related cancers, are increasing globally. So we have a lot of work ahead of us, both at the micro level and all the way up to the macro level.
Well, thank you, Susan. People may be puzzled why it was the White House Conference on Hunger, Nutrition, and Health, and what hunger has to do with it. But, it's an interesting fact that women who are undernourished have small babies. Those small babies, when they're born into an environment where there's excess fat available and excess sugar and sedentary lifestyles, are at an increased risk of getting obese. So we have places around the world, countries where they've never seen obesity before, where there's only been malnutrition. And today they have a rising incidence of obesity, particularly in urban areas. So you'll have small obese undernourished mothers and obese babies in the same environment. And so the healthy nutrition has to start in the prenatal period, during gestation. That's when a lot of the epigenetics is happening in the uterus, and it's an important time for the baby to develop that. And then during breastfeeding is when the immune system develops. So we know that breastfeeding babies, there's a certain bacteria called Bacteroides infantilis, which gets into the baby's digestive system, and the immune system develops around that first bacteria for a period of time before the child is exposed to other foods. And we know that early exposure to a wide variety of adult foods also is a risk factor for obesity. So these are many of the problems. But, the metabolism of the tumor is very much like the fetus, to stay on that theme for a moment, and that is that, when you don't eat and you're asleep at night, about 40% of your genes are turned off, and you're starting the early adaptation to starvation. So by the time you wake up in the morning, there are tiny amounts of ketone bodies that I could detect in your breath with an analyzer, and you've all heard of the keto diet where people starve or they fast. Well, this fasting that you do at night immediately starts that process, and part of that process is taking amino acids from the protein in your muscle to make glucose for your bloodstream, for your brain, and for your red blood cell. So that process starts when a tumor starts to grow as well. And I want to ask Richard some of the practical aspects, because I've worked for a long time on what we call cancer cachexia, which is the wasting of cancer patients. But there's also sarcopenic obesity, where people have high fat and low muscle. And you're in the clinic, you're on the front lines, Richard. Give us a view of what you do for the cancer patient practically.
Great. Thank you. Thank you. Pleasure to be here with this distinguished panel. And I'm a medical oncologist at City of Hope Lennar Foundation Cancer Center, but I also wear the hat of medical director for integrative oncology. And so both of those hats, I get a lot of questions around nutrition from patients I'm treating, recently diagnosed, who might be undergoing chemotherapy, radiation, and surgery. And then we also have to think about kind of the survivorship period. So it's really, two different time zones in terms of thinking about how do we help patients with this idea of nutrition. I really like the idea that David mentioned about precision nutrition. I think that's where we're moving toward. I was talking with Tom Gajewski yesterday, who you heard earlier about immunology in terms of some of these studies that are coming out in terms of how do we use things like probiotics or bacteria that may actually alter the way we look at immunotherapy in response to treatment. I think right now, because we're still studying that, it's really thinking about patients and helping them in a much more practical sense. Because when patients are undergoing treatment, they're going through the nausea from the treatment. They might have just had, patients I see with stomach cancer just had half their stomach removed or the entire stomach. And so it's really challenging in thinking about just the basic nutrition. So I think as oncologists, we haven't done a great job, to be honest, around nutrition. We often have ignored it. And I think some of my colleagues, I hear from my patients, I asked my doctor about nutrition, and they said, oh, just eat ice cream, get enough calories. But I think we're really moving away from that with the data that's growing from other researchers that nutrition is really important. We have to identify malnutrition. We have a lot of dietitians at City of Hope early on. That's our goal, is to help patients even when they're not quite in treatment, but prepare them for treatment, whether it be stem cell transplant or surgery, especially if they have things like pancreatic cancer that affect their digestion. So help them early on through treatment and then through survivorship. And so we may not be able to meet all the goals that are out there because they're going through treatment. So we really have to be practical and work with patients one-on-one, figure out where they are and where can we get them. And also think about socioeconomic issues, social determinants of health. Are there other resources we can really help patients get through that? And then once we get into survivorship, I think we have a little bit more room to be more aggressive with our approaches with nutrition and really try to get them to meet the guidelines. I think there are very good guidelines already out there. The American Cancer Society just earlier this year put out their guidelines for nutrition and exercise, which are really based on a panel of experts coming together, looking at the evidence. And so I think, and ASCO actually has put out guidelines for nutrition and exercise during treatment as well. So I think we really work with these guidelines, help patients, try to get them, meet them where they are, and then try to get them to optimize as much as we can. And then in survivorship, I think we can really move forward and be more aggressive and really help them reduce the risk of recurrence and help them be healthy and recover.
Well, one of my strategies over the years has been to recommend a dietitian to work with an oncologist. Today, the registered dietitians in the United States need to get a master's degree in order to be registered, and I have a joint appointment at USC where I teach in the School of Gerontology. They have a master's degree program for dietitians at USC, and those dietitians are always looking for jobs where they can go. So when I assign a dietitian to an oncologist, two things happen. Number one, the oncologist loses weight. That's number one. It starts eating better, okay? And then the second thing that happens is he's incorporated nutrition into his clinical program. And that's the most important part, because I've seen all the different kinds of, over the years, I've had all kinds of interactions with oncologists. I've worked with a number of oncologists over the years, very successfully, but there are many who say, I'm too busy to do this, I don't have time to do this, I don't want to do this, and I understand that. And so therefore, a dietitian in their office, that person can then spend time and talk to the patient and say, what are you eating? What can I do to help you in a practical way at a ground level? So I think that's very, very useful. And you brought up the idea of survivorship. I think, there's 16 million cancer survivors today. It's a growing population. There's supposed to be 22 million by 2030. So this is a population that we can treat with the same principles we use in prevention in general. So, type 2 diabetes, I call it diabesity. If about 95% are obese people, we can use preventive strategies to do that. And I'm particularly impressed by some of the thinking that Dari Mozaffarian has done on this with regard to prevention in the community and how we can address this obesity, diabetes, actually at the grocery store. And Dari, would you like to share some of your ideas that you've presented in the Nutrition as Medicine? Or medicine, no, I forget, it's Nutrition as Medicine, right? Initiative, go ahead.
Yeah, well, I think first it would be good to just step back a little bit and comment on what we know about nutrition and cancer specifically. So first, there's a lot we don't know. And so I think that we need to have a major new moonshot and investment in nutrition and cancer. And that's not really been talked about in the new cancer moonshot as much as it should be. There was a recent announcement at USDA would be creating a virtual center to combine and bring together people doing nutrition and cancer research, but with zero new resources. So, my optimism for something with zero new resources having a major impact is not high. So I think that's really important. But what we do know for example, from the American Institute for Cancer Research and World Cancer Research Federation, they've systematically reviewed the evidence for how foods affect cancer directly outside of obesity, outside of diabetes. And basically there are 7 kind of major food groups that have probable or convincing evidence for direct effects on cancer. It's low intake of whole grains, low intake of dairy foods, low intake of fruits, low intake of vegetables, those four protective foods. And then on the adverse side, the biggest one is high intake of processed meats, which have been declared a class 1 carcinogen by the World Health Organization, high intake of red meats, unprocessed red meats, and high intake of sugar-sweetened beverages. We've done an analysis, a national simulation model, estimating that those 7 food groups, not eating at kind of recommended levels of those 7 food groups, causes 80,000 new cancer cases every year. And that's about the same number that has been estimated to be attributable to all of alcohol use. And so that's a big number, 80,000 new cancer cases per year. The biggest group of that is the GI cancers, colorectal cancers at the top, about 52,000 new cancer cases a year from poor diet. And then mouth, pharynx, and larynx cancers, about 14,000. So I think that's really, really important. We need to focus on getting more healthy foods to patients and fewer unhealthy foods, in particular processed meats. And by the way, those burdens were larger in Black and Latinx Americans than in white Americans because of worse diet. In terms of how to start to fix this, right, there is no single silver bullet. We are working on a range of initiatives. I think my own personal top 3 priorities, where I think we're going to get the biggest bang for the buck, are around food as medicine, which is the concept of integrating food and nutrition into healthcare. Number 2 is advancing nutrition science and really having major new investment, structure, and authority in nutrition science. And number 3, in advancing nutrition security through our federal programs, not just food security, getting enough calories to people, but advancing nutrition security where the food is also nutritious. Now, in the first category, just as a brief example of what I mean by food as medicine, there's an intervention called medically tailored meals where patients with significant diseases like cancer get medically tailored by a registered dietitian meals, 10 meals a week delivered to them at home at no charge, paid for by their insurance. And multiple studies have shown that this improves health outcomes and lowers healthcare utilization. At Tufts, faculty member Feng Zhang here is leading actually the largest randomized controlled trial of medically tailored meals called NutriCare. It's a multi-center study, including the Ohio State University Comprehensive Cancer Center, Tufts Medical Center, Temple Health Fox Chase Cancer Center, and MD Anderson Cancer Center. And it's going to enroll 300 patients with lung cancer, a very difficult group of patients to enroll, as you know, and randomize them to medically tailored meals plus education or control. And the preliminary results so far, with about half the patients enrolled, looking at some of the secondary outcomes, show that the patients do better and actually have less side effects from chemotherapy in the medically tailored meal arm. And I think Will has thought a lot about how food interacts with chemotherapy. So that's one food as medicine intervention. Ensuring adequate physician education around nutrition is another. But I think the bottom line is there are really clear policy and systems changes that we can make around integrating food and nutrition into healthcare, around advancing nutrition science, and around advancing nutrition security in our federal food programs, SNAP, WIC, and school meals, that would really make a difference.
I think it's an interesting parallel we've been making throughout the day of lessons from the COVID-19 epidemic. And the people who did the worst in the COVID epidemic were people with diabetes, hypertension, and obesity. So clearly that subterranean or that terra firma underneath all of this comes also in our cancer patients. So when they present with us, they may have pre-existing problems. And in the cancer survivor population, it turns out that many of them get a second primary cancer after being successfully treated for cancer. And those second primary cancers tend to be in people over 65 related to diet and lifestyle changes. So I think this is an area where we have an opportunity to really make an impact on angiogenesis and other areas. And Will, would you like to comment on what we could be doing more for the cancer survivor population?
Cancer has been, as we've heard in previous panels, is making its transition from looking at the tumor and throwing spears at it. with the hope of actually knocking out the cancer. In the last couple of panels, we talked about how do we actually support the body's physiology and ask not the question, why did we get the cancer, but why don't we get cancer more often? And that answer actually leads us to understand that, in fact, the physiology of the normal, healthy human body is actually rigged, well-rigged, hardwired to be able to suppress cancer growth. We naturally have the ability to control inflammation, which is a trigger for cancer growth. We normally have the ability to resist blood vessels from growing into tumors. We normally have the ability to repair DNA that would otherwise be mutated to cause cancer growth. And we also normally have the ability to maintain a good, healthy gut microbiome, which talks to the immune system, which is normally rigged to be able to conduct surveillance looking for early, errant, microscopic cancers that we form in our bodies all the time. And so to some extent, the flip of the whole mindset is really how do we help the body do what it normally wants to do rather than simply throw things at the body. And I want to just point out one comment that I think is really helpful because, you know, we're having a policy-oriented conversation. We're talking about a molecular nutrition conversation. Listen, eating healthy is actually quite natural. And if you take a look at the traditional recipes that are delicious, not only the lunch we had today, but if you look at the Mediterranean, if you look at Asia, traditional recipes found in those nations are built of those ingredients that science has now rediscovered to actually have those healthy bioactives that act to activate our body's health defenses that can serve anti-cancer. So for the consumer, we don't need to go into the complex biology. That's a conversation that we have here in this room. For the consumer, they need to understand, you know, simple ideas that you can eat healthy, tasty, nutritious meals that actually are based on time value traditions that taste great. Nobody wants to actually be stuck to a program of elimination. People want to actually go towards the things that they desire. So part of this is educating those of us who are involved with interacting with patients that you can eat things that you love. And I always sort of say, love your food to love your health. That's a good alignment that people can get behind.
Well, you know, I think that, to your point, our U.S. diet has evolved in a certain direction, and Dari related to that a little bit. It had to do with the Depression and the fact that people in the 1930s were malnourished and just didn't have enough calories. So the discovery of certain types of hybrid corn, where it took one man working 1 acre for a year to feed one person, you now could feed 120 people from that 1 acre of land. So we had all this corn, and it was used to make meat to radiate and send to the troops in Europe. And then after the war, you had all these silos full of corn. You turned it into corn oil, into all kinds of margarines and all kinds of stuff. And so it became an area where we had a disconnect. And it's amazing that in 1970, we only had about 10% obesity. People were working hard. There were less, you weren't driving your car as much. You had fewer appliances. You had things, you were working harder every day. So I think as we look at the American diet, we have to accept the fact that there's going to be a period of evolution. And, you know, I think, Susan, as you work in the federal government, you can see this closely working with USDA, but there is an evolution. You know, 50% of all the fruits and vegetables in America are grown here in California. I was one of the original members of the California Avocado Commission. Avocados were not eaten in the Northeast. If you went to Boston, you got green food coloring and mayonnaise for Super Bowl weekend. You know, now the avocados are international. They were grown right here in Brea, California, not far from this Nixon Library. The original tree called the Hass avocado was right here. And from that one tree, it was grafted onto other trees. And now around the world, that one avocado is an international agricultural product. So I think there is hope as people get more educated that we can get away from the brown beige diet and get more toward a colorful diet. And Susan, you're right in the melee. Could you comment on, you know, your interaction with USDA and how you can foster that through FDA? It's food, drugs, and cosmetics, but about the food.
Yeah, happy to. And I think you highlighted so many pieces there. You know, one piece of that is that the food supply continues to evolve, and we can expect a whole lot more evolution in the U.S. food supply in the years to come. And FDA's role there is to try to help support that innovation while making sure that it's safe. And the reason we want to support innovation is to give consumers choices, but those consumers need to be empowered with information to make those choices, and the food needs to be safe. So if you look at, and you mentioned California, you're all sitting out in California. California has had challenges with drought. And meanwhile, biotechnology is addressing new crop varieties that are more resistant to drought. And our role at FDA is to make sure that genetically engineered crops or gene-edited crops are safe. And I think many experts realize in order to feed the global population of the future, we need biotechnology. And so the U.S. is leading many of those biotechnology efforts. In the past, the biotech was really more for the benefit of the farmers in terms of things like weed control. But now what we're seeing is the biotechnology in crops today, many of those are for the benefit of the consumer. So that's, for example, creating genetically engineered or gene-edited crops that have enhanced nutritional value. There's a lot plant breeders can do. And so we do work on that whole issue. USDA and EPA are partners as we look at the evolving food supply through what we call the Coordinated Biotechnology Framework. So USDA's job is to make sure that these gene-edited crops, for example, are safe for other crops. Our job is to make sure that they are safe for humans. And so that's an example of where we play a really important role. But the other thing you highlight is how the food supply has changed so dramatically. We import so much more than we did in the past. The lines of food imported into this country have dramatically changed. And when you look at things like fruits and vegetables, a very substantial proportion of those are imported into the U.S. right now. That gives us more variety, but that also has things we have to think about from a food policy systems perspective for the future. If you look at something like seafood, an important source of omega-3 fatty acids, the long-chain omega-3 fatty acids, more than 90% of the seafood that comes into this country is imported. And so we have critical roles to play as we look at that changing food supply of the future to make sure it's safe and that we can continue to provide food to feed the growing population of the future while making sure consumers are informed about what these products are. So interesting conversations about the evolution of the food supply and what that means for our populations going forward in time.
I think that's a fascinating area for discussion of the preventive values of foods, because as we look at what I like to call phytonutrients, other people call phytochemicals. I think it makes it sound too toxic to the public. I like phytonutrients better. They were originally called phytochemicals because they didn't have any calories associated with them, but they have tremendously powerful effects on cells. And in our laboratory, we're able to show that some of these phytonutrients can be very beneficial in terms of various mechanisms within cancer cells and within normal cells to promote the tumor microenvironment and to promote the whole host in terms of their immune system. And we know one thing, that if you deprive people of food altogether, one of the first things to turn off is their immune function. There are skin tests like for tuberculosis and mumps and so forth. If you look at those skin tests and you apply them to a malnourished cancer patient, they'll have a negative test. As you re-nourish that patient with nutrition, the tests can sometimes come back. So we know that immune function is modulated by nutrition in very important ways, probably through the microbiome. Richard, would you talk about a little bit about what you do to help nourish your patients in an acute basis?
Right, I think those are all great points. I just want to echo William and Dari's point about education. I think even oncologists as well as patients undervalue the importance of nutrition and how much it can really help them feel better, tolerate the treatment better. So I think that's an important area of policy, is educating and really emphasizing the importance there. I think in terms of the microbiome immunology, I think that's a really interesting area, very hot topic. One of our colleagues at City of Hope, Monty Paul, published in Nature Medicine how a simple probiotic with a specific species of bacteria altered the response rates in kidney cancer patients to immunotherapy. And it's probably why I got into Nature Medicine, because it was quite a dramatic difference in responses. Dr. Gajewski and I were talking last night. Apparently there's growing data that certain probiotics may actually have the opposite effect, that when you use them, they might actually have anti-inflammatory effects. And so certain probiotics may actually decrease the efficacy of some immunotherapy. So I think there has to be a lot more research. I think someone mentioned that when a panelist mentioned there needs to be more research put into the area of nutrition. And I think we're finding that, especially in active cancer patients, some of these nutritional interventions may have a real impact immediately, and not just long term, but immediately on the efficacy of the treatments we're giving, such as immunotherapy. So Dr. Gajewski, actually, I asked him this question about, well, what do you recommend? Because he helps treat melanoma patients. So he generally recommends a high fiber diet, plant-based. He doesn't recommend any specific probiotics because he says, there's not enough data. We don't know which one's helpful, which one may not be helpful. So he tends to recommend, probiotics from foods like yogurt. He feels like it's a safe type of intervention. So really kind of following the basic American Cancer Society guidelines on plant-based, more plant-based, fruits and vegetables, avoiding red meat, processed foods, and then thinking about more fiber seem like important strategies at this time.
Well, it's interesting that, the fruits and vegetables also have the fiber as a prebiotic as well as a probiotic. You may not know the difference, but probiotic's a live bacteria. Prebiotic is something the bacteria eats that then affects the bacteria's growth. And fibers and phytonutrients, things that they're colorful compounds, are actually prebiotic as well. And they produce things, the bacteria, called postbiotics, which then can affect your immune function and other functions in the body. So as we start to consider the overall effect of nutrition in the cancer patient, we have to have some guidelines, and I think that the public health gives us those common sense guidelines. I think one of the hesitations is people have become so obsessed with nutrition and multiple supplements and so forth that they have ignored their healthy nutrition. So I think the common sense guidance that Will was coming back to is really important, to eat your traditional diet. Go ahead, Will.
Yeah, no, I think there's another aspect to add to this we haven't mentioned yet, which is the importance for trusted sources of information. And I think that's something that, academic centers try to do. The FDA, obviously, and the government is a source of that. But if you really take a look, just search nutrition on the internet or on Amazon, and you'll see all these claims and products and thought leaders that are out there. And honestly, the consumer has no idea who to trust. You're looking at a panel and an audience of people that are probably much more educated than the typical consumer. And yet, something we learned from the pandemic is that if you go with the science, you got to watch out for misinformation. And then if you take a look at an area like food, there's a lot of polemics out there as well. There are people that believe in this or believe in that. And so where is the confused but earnest consumer going to get their information? That is something that I think we all need to collaborate on. I'm advocating for sort of an annual conference of physicians that is not necessarily nutritionists, but really allow us to address the confusion that we have in managing patients and really invite the people that are the true experts and then make that type of convening publicly available so they can see how we as medical scientists have this conversation so we don't have the answers clearly laid out yet, but this is a journey to find those answers.
Well, it's certainly a complex area. We've talked about everything from public health down to the endothelial cell generating a protein that stimulates angiogenesis. So I think we've got time. If there are any questions from the audience, I think we could take those questions now and be happy to take those. You must want to have some information about nutrition. There you go. We're going to ask Anna Barker. There we go.
That was really great. I learned a lot. Thank you all. I had a question for our FDA colleague. I just read this morning that my colleague, our commissioner, actually said today that FDA was going to start paying a lot more attention to food. What do you think that's going to translate into?
I think you're on mute.
I don't know if he's checked in with you on this or not. So.
Oh, yes. We've been discussing this a lot. So what I would say, Ann, and it's good to see you again, is when it comes to food, we do have challenges on the resource side at FDA. An example of that my budget in the Center for Food Safety and Applied Nutrition doesn't have any user fees. And on the medical product side of FDA, the vast majority of the budgets are coming from industry paying those user fees. So what it means is we need resources on the food side of FDA, and a recent report that came out really highlighted that. You may be shocked to know that the center that I lead has the same number of employees that we had in 1978. But the complexity of the work that we now have to execute with the increased number of import lines, with the technology that's coming. We recently completed our first consultation on cell-cultured food. So it's just the complexity continues to grow, but the number of people I have has not grown. And so we are hoping that a big emphasis will be put on the food side of FDA, because from my perspective, we touch every American every single day, and we would like to see a greater emphasis. I will note that the White House conference agreed with us and actually put a request in the deliverables of the national strategy to better resource the center that I lead in the area of nutrition. We've also spoken about nutrition research. Nutrition research is foundational for all of our work. And many people have pointed out that we really could use more research, fundamental foundational research in nutrition. So one of the things we have been doing is working with NIH to try to make sure that when NIH funds large cohort studies like All of Us, or like the ECHO cohorts that look at people at the earliest, the prenatal period, what the mom is eating, what the infants are exposed to. We'd like to see those cohorts well-resourced to collect high-quality dietary data, so that we can do the types of nutrition genomic interaction research that is so important. All of us, as you know, is investing in significant genomic information. We need to augment that with great exposure information around nutrition, and that will be the high-quality data that we need for precision nutrition going forward. So I can tell you I'm all over Washington advocating for research and resources to put a bigger priority on nutrition for all the reasons you've heard here today.
You know, it's fascinating. The nutrient database developed by USDA, they date back to 1973. And many of the foods that are in the nutrient database are no longer on market shelves. And I'm working with a nonprofit that's working on an open source data for everyone to use in nutrition science, so we know what's in our food supply. So nutrient analysis is kind of the foundation of precision nutrition. If we don't know what foods people are eating, we can't really speak to all the other aspects of the genetics, the epigenetics, et cetera, if we're starting off with the wrong information. So I think that's a really important area, and I think it's interesting because the food industry in many ways is larger than the drug industry and could certainly afford those user fees. And so I think it's an issue of assuring people that this is not going to, I know early on when I started talking about nutrition, people, oh, you're going to ruin people's confidence in the American food supply. And I said, no, that's not the issue. You're going to get a better food supply, and I think that the USDA needs to understand that we're working to make things better and not to stop things from happening. And there'll be a natural evolution over time to a better, healthier diet as we all work together on this problem. And that will ultimately have an impact on cancer. I take the example of liver cancer, which originally was due to moldy peanuts or whatever, today is overwhelming, or hepatic viruses, but today is overwhelmingly due to fatty liver, which affects 40% of all overweight and obese patients, and then a small number of them get liver cancer. So we can see the impact of foods, yes, toxins, yes, but also obesity and overweight and sedentary lifestyle. Okay.
David, can I add something to this comment?
Sure, go ahead.
So, as we talk about the overlapping disease conditions that are relevant here, cancer, obesity, diabetes, fatty liver, the gut microbiome, and the need for more research, we did a systematic analysis of nutrition research and published a detailed paper on this a couple of years ago and shared it with the government, with the U.S. government. We're not going to get to where we want to go, at least not in the next 10 years, unless we actually fundamentally change the structure and authority of nutrition science. And what we believe is the top recommendation is to create a new National Institute of Nutrition at NIH. And so the same way the National Cancer Institute was created by a coalition of a small number of people who went to Congress and said it's time and really dramatically transform cancer research, it's time to do the same thing. It has to be congressional action. The administration can't do it because of statute. So Congress has to create a National Institute of Nutrition. The budget should be additive to all other research funding. It shouldn't take away from any existing projects going on anywhere. We don't want to silo nutrition research, but $1 billion or $2 billion a year for a National Institute of Nutrition would be very, very sensible. The estimates are that poor diet costs our economy $1 trillion a year in healthcare spending and lost productivity. So 1/1000th of that a year, would be a very, very good investment. And so I really want to call on everyone in the room to speak to your elected representatives, talk about this, that Congress has heard about this, and it is possible, especially with the momentum of the White House conference, to actually get Congress to act and create a National Institute of Nutrition.
Yeah, absolutely. I think that would be a tremendous, great support in the room also, Dari. I think, that it's interesting, when I was a director of a clinical nutrition research unit, actually related to the NCI, and there were 8 of them in total. And I remember having discussions at the National Institute of Diabetes, Digestive Kidney Diseases, and I would raise the issue of obesity, and the director at that time, well, we know how to do research. Don't tell us about obesity. It's just don't tell me about it. And then there came the mark on obesity. There was funding available. All of a sudden, the next meeting, he goes, we have always been enthusiastically working on obesity, So I think that money will move the needle, and I do think that your idea is a good one, and it would be very important. Yes, Andy. The challenges of, I want to get Andy von Eschenbach, former director of the NCI and the FDA, wants to make a comment, so let's hear from Andy.
I'm sorry, I just wanted to follow up on your comment because it's incredibly important. To get another institute at the NIH is going to be a formidable challenge, but ARPA-H, the Advanced Research Programs and Agency that is now being created for health, is in place, and its agenda is just starting to evolve. I would really suggest that in addition to efforts about another institute, you really focus on ARPA-H and getting nutrition into that agenda, because it's there, it's been approved by Congress, funding will come, and you ought to be at the front of the line.
Yeah, ARPA is an amazing agency for advancing research, and a lot of the breakthroughs on the defense side have come through ARPA, and now with ARPA-H, that same innovative spirit toward health advances that we need in nutrition and cancer can come. Okay, other questions? Over here.
Hi, good afternoon. I'm not a researcher or a physician. I'm actually just a docent here, but I'm the son of 2 former cancer patients. My father passed away 2 years ago from it. My mother passed away many years ago from another illness, but she had it at the time, it's breast cancer. My question is just how closely tied does the data indicate sugar is in the diet to a broad range of cancers at this time for just regular people who have maybe not the best of diets and not the worst? Thank you.
Sure. Well,
I'll tell you how I answer that question. Most people are perfectly able to process normal amounts, moderate amounts of sugar, especially the sugars that are naturally found in fruits and vegetables, as an example. And it's not a problem. And there are some people that really, sort of state you should stay away from all fruits and vegetables. That's way kind of like over the boundary in terms of what the evidence shows. In fact, quite the opposite. So what I would say is that what's clear is that added sugar, a lot of added sugar, which in this country we eat a lot of, it's present in our industrialized food, that actually has all kinds of domino effects that can actually lead to chronic disease, including cancer. Now, there's, for every urban legend or popular idea about sugar or soy or some other tomatoes with lectins, I mean, there's an urban legend surrounding it, and then voices that actually propagate these, really false messages. They're false totems. And what I think we should understand is that if you have diabetes, you need to actually, or a metabolic syndrome, you do need to be careful about the amount, the volume of sugar you eat. But if you get it from whole plant-based foods, you're going to be just fine. I think that's the key thing. The other thing I would actually say that's really important is that there's so much emphasis, and I think a little bit of the discussion today has touched on this. There's an interesting tension industry is motivated to spend money to tell people what to eat, oftentimes the food that's not so good for us, as research known. Physicians, on the other hand, have been trained to kind of tell people what not to eat. And so we kind of demonize foods as well. And so you've got two voices that really are very confusing for the consumer. What we do need, and I think this is what a new institute or ARPA-H and collective research could do, is to focus on not so much what we should remove. I think the bad guys are pretty well known. And we don't need more voices to say what we shouldn't be eating. Human nature abhors deprivation, and nobody wants to hear another bad word about food. But what everybody wants to know is what I should add. What should I add to my diet? And this is where a medically tailored meal for cancer patients could be incredibly important. And it's going to be personalized because everybody's different. Everybody has a different metabolism. Everybody has a different backstory, comorbidities. And that's really where you can't generalize something like sugar, although that's a natural question.
Well, I'll just add quickly, in clinic, and just like where you're saying, William, is that you'll see patients come to my clinic and say, well, I heard sugar is bad, and we know, I'm not going to eat any sugar, I'm going to go on a ketogenic diet. And when patients do this very kind of extreme diet, they come back 2 months later, their hair is falling out, they've lost 50 pounds. I said, wow, what happened? Well, I'm not eating any sugar. I said, well, look, that's not, they've gotten misinformation, they've gone too extreme, and it ends up causing more harm. And then I have to convince them, look, let's come off of that, let's come back to a more balanced Mediterranean style diet that meets all your needs, not one of these kind of extreme fad misinformation-based diets. So it can be quite harmful. So I appreciate all your comments. I agree with everything, and it's really a balanced approach and going for kind of natural foods and avoiding the processed added sugars and those things.
Yeah, the problem with nutrition is everyone who eats is an expert on nutrition. That's the problem. Go ahead.
Sarah, can I add on, David, just on the added sugars?
Sure, then we'll wrap up very quickly. Thank you. Go ahead.
Yeah, it's just, and Dr. Lee spoke to it. You've got fruits that contain sugar, but when you're eating those fruits, you're eating them in the context of a nutrient-dense food, right? And people understand that. Same thing, lactose is a sugar. And so when you're consuming those dairy products, you're consuming a sugar. And so the critical need was to differentiate what are added sugars from those that are really coming from these important food sources that we've heard are really important for cancer prevention and overall health. So that was the criticality of getting added sugars on the nutrition facts label for the first time in 2016 with a daily value. And the importance there is we worked with other federal partners, including the National Cancer Institute and their dietary modeling group, to say that that a healthy diet can incorporate some amount of added sugars. But if you have too much, you're either going to overconsume calories or you're going to miss out on key nutrients that you need. And so it's a critical thing that we need to work on in terms of education. That's the importance. And we've worked with health professionals. We've produced videos and things to educate health professionals on how to work with patients on things like the nutrition facts label, including added sugars. But just a few comments from a regulatory perspective, what we've been trying to do on the added sugars landscape.
Well, thank you very much, and I want to thank all the members of the panel. I want to thank the audience for their attention and interest, and we'll wrap it up right there. Thanks so much. Have a great afternoon.
Ladies and gentlemen, that concludes our third panel. Our fourth and final panel on technology and medicine will begin at 2:30 p.m.
Ladies and gentlemen, I invite you to take your seats as we begin our final program. As we close this year's Nixon National Cancer Conference, we'll explore the intersections and applications between technology and medicine. As both fields continue to evolve and advance rapidly, a successful pairing of the two could lead to promising and indeed revolutionary results. Stanbridge University is an award-winning healthcare-focused system and a leading innovator in ed tech. The first to introduce the use of synthetic cadavers on the West Coast and large-scale virtual reality in foundational science training, it is leading efforts on the use of AI-enabled social robots in a variety of pediatric therapy treatment settings. It's now my pleasure to introduce Yasith Weerasuriya, President of Stanbridge University, to introduce our final program.
Thank you, President Byron. Good afternoon, ladies and gentlemen. Would you please join me in recognizing the youngest president of the Nixon Foundation, an accomplished orator, and his talented staff for their hard work in successfully hosting a very successful second annual Nixon Cancer Summit. To introduce our final panel, I thought I would harken back to a panelist in our beginning panel last evening, Dr. Ferrari, a mathematician, a physician, and a musician, may I say extraordinaire, who brought humanity to a room full of science with his excellent singing talent. His words struck a chord when in describing the trait he thought best captures a predicate of success. He said it's those who have grit and resilience and the ability to rise from failure and pursue their dreams again and again. And in the race for a cure for cancer, he said that resilience and grit was driven by human moments, perhaps the illness or the passing of a loved one. He reminded me of another mathematician, John Nash, who was immortalized by Hollywood in the movie A Beautiful Mind. In his penultimate moment, receiving the Abel Prize, he said that for all of his journeys in math and science, it all came down to human moments and love. So to introduce the moderator of a distinguished panel of physicians and scientists who would speak about the latest in technology and medicine, I thought I would skip reading his extraordinary list of accomplishments and simply ask him two questions as an introduction. As a member of the healing arts profession and physician, what was a seminal human moment that he remembers? And Dr. Pistes answered that it was over a decade ago when he had a patient who presented to him a Jehovah's Witness that had been rejected by all surgeons previously and had no hope. And he said he and his team took on the patient at MD Anderson. And he said that after a 14-hour operation, that successful surgery is one such human moment for him. And as a second question and a segue to the panel today, I asked him what technology he thought would move the needle in medicine in the days ahead, to which he replied very carefully that it is all of the different chords of artificial intelligence and data science that he believes where we have hiding in the freezers and the servers today, the answers that data science and AI could unlock, answers for the future. Please join me in welcoming a distinguished American teacher, surgeon, and accomplished leader in medicine, Dr. Pistes, who will introduce his panel.
Thank you very much. As usual, we've saved the best for last, and this is an opportunity for us to come together for an outstanding panel discussion. And so with me, immediately to my left, is my good friend and fellow surgeon, Bill Kantz, who's a research scientist at Grail and has a long-standing history with the American Cancer Society, as well as outstanding service in a number of different academic medical center environments. Bill, do you want to say a little bit more about your background and your current role, and then we'll go to the other two panelists.
Sure. So as Peter said, I've done cancer surgery and cancer research my entire career, kind of trying to bridge the aspects between the lab and the patient and back. So as I've moved forward working on invasion metastasis during the career, studying focal adhesion kinase as a central molecule, as I went through, I realized I wanted to get to the next level of where we're going to be going. Where's the field taking us? I went to the American Cancer Society, and I met my colleagues at Grail shortly thereafter. Worked with them for several years. Impressed with the depth of their science, the commitment to science, and saw how the early detection is so critical to the next phase of what we do, and how technology is going to be such a big part of that. So delighted to be here with you all and join the panel. Thanks.
Thank you. Now to our virtual teammates. David Agus is founding director and CEO of the Lawrence Ellison Institute for Transformative Medicine and a CBS physician, I believe. So, Dr. Agus, can you tell us a little bit more about yourself? Do not describe yourself as just a medical oncologist, but give us some insights into where you sit right now at the interface of technology and medicine.
Apologize. Now you can hear me. I am a medical oncologist, and you figure after 3 years of Zoom, I would be better at that, and I apologize. And it really goes to the fact I focus on using technology for disease, and I can't use technology to speak. So it's we had an institute that cares for patients. I had a full day of clinic today. And as well develops therapeutics and technologies to benefit patients. And I also chair the Global Health Security Consortium, or co-chair. So we do something on a global basis, again, to try to understand how to better disseminate the advances we have in cancer to people across the globe. I'm a believer that many of the answers are going to come on the data side, and that we've really hit a lot on the biology side already. And the real challenge now is to look at our outliers, whether it be the amazing responder or the person who doesn't respond at all, and from these outliers, understand what's better. My field has been very good at the mean historically. Now we have to widen our view.
Thanks so much, David. And then finally, Dr. Peng, who comes to us as a physician and a researcher and director of product development at Verily. Thanks so much for joining us. Give us a little more background about where you sit right now and a little bit about the company.
Yeah, thank you for having me. So my name is Lily Peng. I am a Director of Product Management at Verily. I'm actually a little new to this role. I just started about like a month ago, and bulk of my work actually was done at the sister company, Google. You may have heard of them. And a lot of my role there was to apply AI to healthcare. On the research side. And what that meant was, there's a saying, it wouldn't be called research if we know what we were doing. So a lot of that is actually learning from a lot of failures and learning not only about how to apply technology to the healthcare space, but how do we translate that technology into the real world, into real world impact for patients. And one of the reasons behind my move to Verily is that a focus on building those products and accelerating the translational piece. Because what I've noticed from my 8 or so years of just working with the Google team is that we train models every day. Your phone, you can like type in the name of your dog and you can find your dog. So AI is powering consumer experiences all the time, helping consumers and patients, all the time. And so how do we accelerate that translation into the healthcare space? And, not to give things away, but having great data, I think, is a big piece of it. And I know that I'm looking forward to talking about that a lot more.
Well, let's open up the discussion. Probably start with you, Lily, really looking at the reality of how ready is healthcare for artificial intelligence. We see it in all of our daily lives with Siri, with Alexa, with other aspects of autonomous driving, to give another example. Is healthcare ready for AI, or what are the limitations that you see about deploying AI for patient care or for predictive analytics?
Yeah, I feel like we are both so ready like we need this yesterday type of ready, but also so far out. And what I mean is, we have seen lots of real promise and applicability on the research side, right? I used to work a lot in applying AI to medical imaging, right? And we show how well AI can actually handle images and actually give you really insightful, great insights from images, great for rote tasks, of which we have a lot in medicine. But then I think the process of translating that technology into real world impact means generating evidence. This is not like a consumer space, right? Like if the product malfunctions, there are real consequences, right? Real harm done to patients. And so we need to generate evidence that whatever technology we have, whether it's AI enabled or not, is effective. And so, the need is there, there's clear demonstration, but the translational path, I think, is not quite there. It's not fast enough. I mean, it's there, but it doesn't handle the volume and the cadence that we can improve. Oh my gosh. Healthcare. It got really dark really quickly. Technology just decided that was not in this room. Anyway, so let me just finish that part and to say that like the process of translation is, I think, where we need to actually put in a lot of muscle. And so it's less about AI itself, and it's about the process of translating technologies, period.
Great point. Alexa, turn on the lights. David, your thoughts about that?
You just turned on my thing, okay. By the way.
We can see how well it works.
Yeah.
If we unpack a couple of the use cases that Lily was pointing out, pattern recognition. If we can really have YouTube find videos with cats in them, why can we not really readily deploy AI methodologies or machine learning for radiology interpretation or pathology interpretation? These are fields of medicine associated with pattern recognition.
Yeah, I mean, the bottom line is, no offense, I don't care about radiology or pathology interpretation. I care about improved patient care, right? I mean, that's going to be the key thing with all of them. That's the killer app, if you will. And so the problem is that we don't have structured data. Electronic health records are a bag of words. And to nobody's fault, that's where they were developed a long time ago, and they really haven't changed the underlying technology. And so bags of words are hard to put and create into structured data, and you need structured data in order to develop insight with machine learning or artificial intelligence. I will tell you that we all know, right? Some doctors are great, some doctors are good, and some doctors are average. Right there, the way that distinguishes a great and an average is better pattern recognition. I tell you, I look under the microscope, at all of my patient slides, and I can tell, even though that, the pathology says this, it's going to be more aggressive than they say. And it's just pattern recognition. So if that's the case, I know machine learning AI can do it better than me and can make every doctor great in this country. We talk about making new advances in biology and new drugs. I really believe if we can develop structured data across the country with electronic health records and with AI, we can equal care across the country, and we will probably save orders of magnitude more lives than if we developed a half a dozen new therapeutics, and it will be orders of magnitude cheaper. So that's going to be the goal now. And I think there's going to be a transition. Remember, it was a feature, not a bug, of electronic health records that you couldn't get data out of them. That has to change. Every patient is in charge of their own healthcare data. It's not their doctor's data. It's not the institution's data. It's a strange artifact that we stripe data by institution. Why is there the Mayo Clinic data set, the MD Anderson data set? That makes no sense. There's the United States of America data set. And when you think like that, we can then start to have structured data where we will all benefit. If I said, hey, listen, give me all your credit card records, give me, all of your, financial records, and I'll consider giving you a home loan. You do, even though you don't know who I am. If I say, let me use your healthcare data in a privacy protected way, right now people say, oh, it's my healthcare. We have to change that attitude. Together with the structured data, we can radically improve healthcare.
I'm going to ask you one follow-up question before I go to Bill, because you're really hitting on the key point. We have poor quality source data. We have bags of words. what is the strategy for moving to structured data in individual institutions or medical practices or at large scale?
There is no cogent strategy is the bottom answer. But I do think you're going to start to see organizations like CMS require more structured data for reimbursement. I think we saw it at the FDA. It was the first federal agency to require standard data elements for a submission, and that was fantastic at the time. I think you're going to see the electronic healthcare companies, Cerner, Epic, and others, who are going to start to convert from this legacy software, which is literally like MS-DOS from the 90s or Windows 95, to modern software in the cloud, where we will have standard data elements and structured data. I think it's going to be a new revolution. We saw it with Oracle, and obviously, Larry Ellison is a big donor to my institute in all full disclosure. That being said, Oracle buying Cerner, I think, was an enormous step forward, and you're going to start to see, advances now where data is going to be freely shared, and we're going to have the ability to really make an impact. You can imagine every academic, every person in a company who works in machine learning and AI is dreaming of getting structured healthcare data to make an impact, and it's going to happen.
We can really see it, and just to give you the MD Anderson example in terms of what we're doing is moving to synoptic reporting for radiology reports, for operative notes, and for pathology reports. And if we can put those data sets into synoptic format by still allowing a radiologist, for example, some freedom to dictate free text, but key elements recorded synoptically, we have a way in which we can use that for research purposes. It's what we refer to internally as a first mile data strategy rather than a last mile data strategy. Most of us are familiar with
that strategy, but I showed that strategy that you were doing to a bunch of Silicon Valley guys and girls, and the first thing they said is, "This is amazing. Why didn't they do it 20 years ago?"
That's because we had too much freedom and too much autonomy and no focus really on how to provide high quality first mile data and a reliance on what you would describe as last mile data strategy that we all knew as medical students and fellows. Okay, let's get into diagnostics. GRAIL, a blood test to diagnose cancer. This is something that we've all been looking forward to. The opportunity to avoid a bowel prep and a colonoscopy, I know is something that's on my to-do list. So help us to understand what the technology is and what the science is behind this.
Sure. So let's just sort of level set first. What's the why behind it? Why do we need to have early detection? 70% of the cancer deaths in the United States are from cancers where there's no screening test. 70%. And when you look at the numbers, the demographics, the baby boomers aging, we know that age is one of the biggest risks for cancer. It's overwhelming. 10,000 baby boomers turn 65 every day. So we have individual screening tests that we've been using that aren't sustainable. So we really need to move to screening individuals for cancer rather than individual screening tests. So that's the why behind the early detection. So how did this actually start? It's an intriguing story. A scientist at Illumina was looking at cell-free DNA in the blood of pregnant women and looking for abnormalities in the fetus that could be detected from the fetal DNA. And she found tumor DNA in some of the mothers. And they went, whoa, and people are right, yeah, prove it. Every one of those women had cancer. So she found circulating tumor DNA, cell-free DNA. So it was the idea that tumors themselves are shedding DNA when they set up shop in someone's body. So that was about, in 2016 when GRAIL spun out. And the technology is not just sequencing DNA. We've heard a lot about DNA and we've heard a lot about regulation and so forth. We have to get beyond just DNA sequence and mutations and look at how that DNA is regulated. How's it turned on? And it's methylated. That's what switches on individual genes, the DNA methylation, it's transcriptional control and so forth. So the first piece is actually sequencing the methylated DNA in a blood sample. That's the first part of the technology. It's called bisulfite sequencing, but you can sequence these little islands, and we sequence about 1.1 million in every sample. 1.1 million. So there's a technological advance right there. How many of you all remember manual sequencing? How many ever poured a manual sequencing gel? Yeah. You know where you had CATG, your fingers were CATG and you were entering in the computer? 1.1 million. So how do we analyze that? And that's where the AI comes in. So we've been able to define a common cancer signal that's over at least 50 cancers through AI machine learning of the methylated DNA. And we can also, by looking at specific CPG islands, tell the cancer site of origin in over 90% of the cases to allow directed workup of the positive tests. So that's the technology. We have over 330,000 people that have had the test in clinical trials, so very deep research commitment. Over 50,000 have taken it commercially because it's been available now for a little over a year. Just quickly to tell the final, sort of the latest results, which we presented at the European Society of Medical Oncology in September. This was a study we called Pathfinder. About 7,000 people were enrolled to see how it was implemented. That was the endpoint, the implementation. So here are the three things. We doubled the number of cancers detected from just ordinary screening. So we doubled it. And 70% were those that didn't have screens. 2, they were deadly cancers. There's some worry of, oh, you're picking up these cancers that will never matter. No, these are deadly cancers: pancreas, liver, endometrial, osteosarcoma, head and neck. And about 50% were stage 1 and 2. So our key is that we want to see across all development of multi-cancer early detection, we want to see that stage shift to earlier stages. So that's in a nutshell, but I spend a lot of my time on the data piece. I mean, how can we link the data? It's such a critical piece of getting real-world data that's regulatory grade for the FDA.
David, your thoughts about this? I know that you take care of patients. How do you think patients are going to perceive this? And what are the limitations of this in a situation where we're rolling out a test, where we're trying to define using real-world evidence, false positives, false negatives, and at the same time, work on the coverage issues?
The science is interesting. It's still nascent. It has a ways to go. And I think that's the hard part now. Yes, it is available to patients who are rich, who can buy it. And what I've seen are things that can be scary. I have a dozen patients that have had significant operations or procedures that probably didn't need it. You know, there's one argument that stage 1 cancers will always be stage 1 cancers. You have to show that finding these early matters. And I think that's very difficult in many regards, rather than have life-altering surgery. At the same time, you know, when you take 100 people with known cancer, you know, only a small fraction will turn positive on the test. So many people take the test, don't go to other screenings, and end up, we've seen several with metastatic disease because they were, quote, false negatives on the test. And so I think it's very difficult. That's not a fault of the test per se. It's a fault of the people who do it, who are doctors who have no idea what it is because it's being sold by community docs, basically. And they don't know how to truly give informed consent to the patients to understand what they're going through. And so the notion of having a nascent test that hasn't been shown to affect outcome available to people who can afford it, to me, doesn't make a lot of sense, and is being misused in that regard. That's not to say it won't work and won't be a fantastic advantage in the future, but we really have to do the studies first before you put that out there and really cause tremendous stress or potentially tremendous procedures in an individual patient.
Bill?
You know, and David, those are great points. I mean, we do not want this test to cause undue psychological anxiety, and of course we don't want unnecessary procedures, and that's not, and we haven't seen a lot of that. Let's look at the data of what we have seen so far. The false positive rate is 0.5%, so 5 out of every 1,000 tests, and the positive predictive value is 43%. So if the odds of having cancer with a positive test are 43%. Put that in context with mammogram, lung CT, they're well under 10%, 5 to 7%. So we have looked even at the people with false positives. We've done patient-reported outcomes in the trial I just mentioned. And over 80%, even with the false positives, over 80% of them are satisfied and felt that the anxiety level was not excessive. To your point about trials, absolutely. We recently finished enrolling the NHS Gallery trial. The National Health Service in the United Kingdom, we enrolled 140,000 patients in 10 months. Fastest enrolling clinical trial in history. It was faster than the polio vaccine trial. So and it's a randomized trial. So people got blood drawn, but it's randomized who got the test, who got Gallery, and who did not. So those results will be coming. I think you make a good point that we do.
By the way,
need to see
the COVID vaccine trial enrolled 120,000 patients in 3 months. So.
Well, then point is well taken. But it wasn't a cancer trial, maybe. Well, neither was polio. So good point.
Lily, what are your thoughts here about how to integrate the data science with the technology?
Yeah, I mean, I by the way, I'm really enjoying this debate, and I think, at the end of the day, one piece that, I hear that's both a pain point for both people who are developing these innovative technologies and patients that could benefit but also could be harmed is that really we don't have a way to generate evidence nearly rapidly enough to show whether or not, hey, overall, because every time you take a test, there's always that risk of false positive and false negative, right? It's always this risk that you have to take. And without the right data, you're not able to really, inform that decision. And then there's a big difference between how things are run in the pristine condition of a clinical trial where you have all of the support, right, and monitoring and all that. And then what happens in the real world where you have folks that are not as well informed about the test, even clinicians, right? And then also patients who are also not as well informed and do not have as much support. So I think there's two pieces. One is how do we rapidly generate evidence? The NHS is a great place because of all the integrated, it's still one system, and yes, they're different systems, but they can integrate and talk to each other. And that's why, with the COVID work, they were some of the first to find out about, therapies that worked, right? Like dexamethasone worked, and they were able to do things rapidly because they were able to merge research and care together. And that's one piece. And then the other piece is like practically how do things play out in the real world? And I think the framework is actually thinking about those different types of ways that we're gathering data and how do we make sense of all of that and gain insights from all of that. I don't know that I actually answered your question, but because this is like these big themes, I think it's worth kind of highlighting around these two different areas where I think we need to do more investment.
Excellent points. A couple of things I want to drill down on a little bit. The first you alluded to, and that's the role of patients. How can their voices be incorporated into the generation of solutions here, and what David, I think, accurately describes as use of their data? Lily, what are your thoughts there?
Yeah, I think this is a key piece, and I think other folks on the panel have alluded to this, is that participants or patients, we keep screening for this type of cancer and that type of cancer, right? It's like, well, I'm at risk for all kinds of cancer, right? Well, maybe not prostate. But, I'm at risk for all of these kinds of cancers. Like, how do we, screen people for the thing that they care about? They care that they don't get cancer, period, or that that's found early, period, right? So I think that's one super interesting concept. Another concept is that patients generate lots of data outside of care and outside of research, right? How do we capture those experiences, right? And also, how do we capture the experience of going through a therapy? So let's say I'm on a drug, an antidepressant, and I'm actually losing sleep, and sleep is actually really important to everything else. But we don't know this because we're not actually measuring the thing. Like, you don't measure sleep in a care episode in a patient visit, right? So how do we measure all of these things that contribute to well-being for a patient? And I think all of this, in order to do this, there needs, now I have a kind of a little strong of opinion, which is there needs to be the ownership of the data needs to centralize around the patient, right? Because they are the connection points for all of these different sources, whether they are a Mayo patient for a certain period of time or, Sloan Kettering for another time, they are that central organizing force for all of it. I understand why it's so hard to do that, because the patient itself, like the patient himself or herself, they don't have an IT team like Mayo does to build a system that's organized around them. But I think technology has made it possible that, like, you have a Google account, you have a Facebook account. We actually do centralize around the consumer or the user or the patient. So it is possible. We just have to figure out how to implement in the right way. And then we also need the other stakeholders, to buy in, right? Because, like, it's incomplete if we don't get the care data. It's incomplete if we don't get, if you're in a trial, the COVID trial, that data. So I think we need to agree that the patient matters, the person matters, and that we need to try to get more and more people who believe in this and to join in. I don't think everyone will agree all at once. I don't think that's pretty quixotic. I think you have to convince people to join over time.
Sounds like a religion.
It is. It's a bit of a cult. You got to believe it. And if you believe it, it might just happen.
During COVID, we were part of a team that developed a program called vSafe, where every person in the country had the opportunity of clicking on a QR code and entering the side effects they felt. That data enabled us to have data on pregnant women and breastfeeding women who were excluded from the clinical trial. And so several months after launch of the vaccine, we were able to get FDA approval for them from this. And it's really the first time that we collected data after a drug or a therapy, or vaccine in this case, was approved by the FDA. The notion that clinical trials stop when they stop arbitrarily at a certain time point makes no sense. In the oncology world, 60 to 70% of the drugs we use are, quote, off-label, and we don't collect any data. So the idea that real-world evidence is inferior to stuff on a clinical trial, again, is something that has to change. Every patient should be given the opportunity of being part of the cure or a way of improving the treatment for a disease.
How do you get the health systems to buy in?
They have no choice. It's not the healthcare. It's not the health system decision. It should be the patient's decision. That's the point.
It should be. That is exactly right. But that's not the way it plays out. As we try to get real world data, it's like, no, you can't have the data. Can a patient do exactly what you described with a Gallery test? No, they can't. Because the systems will not let that happen in some cases. So I think that's because it's our data. So how are we going to get past that? Because that's such a critical component to the real world. We have to be able to look to see the real world implementation and what happens. We have to get those data. I totally agree, but it's hard. I think it's harder than doing the AI, to be honest with you.
David, your thoughts on how to optimally integrate patients. I think we agree on the notional concept that the data belong to patients. But how do we help them to understand that, and how can they opt out in the future from specific uses of their data?
There's this ruling that came about called HIPAA, the Health Insurance Portability Act. Sounds very sexy if you think about it. It's like taking a suitcase somewhere. But in reality, it was meant to protect the safety of patients and the integrity of their data. Unfortunately, it's been interpreted. It's not the law, but it's the interpretation of a hospital's doctor. Oh, it's HIPAA. We can't touch the data. We can't play with that data. And I think there really needs to be a new sense of patient bill of rights. And I think at some point we have to go that direction. The patient has a right to their data. The patient has a right to be part of research, extra institutions. So it's not just at that hospital or that institution they're at. And I think that's where things are going. You're going to see it. The UK is the worst example there is, right? In the UK, the hospital doesn't own the data. The National Health Service doesn't own the data. The individual general practitioner owns the patient data. And it's a remnant of something from 25 years ago. And that has to change because it literally makes no sense. That individual practitioner can sell their patient's data, but the patient can't have access to their data. That's not right. And so I do think we're going to evolve to a point, and I think there are a lot of meetings coming up where we're going to get toward this patient bill of rights, and it has to happen. Technology has given us the ability, and it's really an inflection point, right? We now have the cloud to store things, which is remarkably secure. We have the ability of privacy protecting the data like we never have before. So technology is going to enable us to liberate this data and have it used for public good.
Without getting into too much detail on the technology, do you see like cloud-based solutions or blockchain technology as ways in which individuals can retain ownership of their data or control of their data in ways maybe that they don't imagine today?
I have two words for that answer. Hell yes. If you look at it right now, right, my hospital, I'm not going to say which one we're affiliated with, is a small hospital. It's got some IT people who are really well-meaning, but no offense, they're not very good. And you think they have the ability of protecting their patients' data? How many hospitals have had ransom, payments this year because somebody has taken over their data? Once you transition to the cloud and have literally the top security experts in the world maintaining our healthcare data, it's going to be much more secure than it is now. And technologies, whether they be blockchain or other technology that enable you to have custody of your data from multiple sources, are available today and will be implemented hopefully in the next year or so. I hope grand scale.
Terrific. I want to move to the talent dependency associated with this future state. We've heard in the course of this conference how mathematics and biology are being merged. Computational biology is really a field. Biologic phenomenon can be described by mathematical equations. We are competing for talent with industry and with other sectors, and we can't, at least on the provider side, we can't compensate at levels that attract data scientists that get paid millions of dollars at Google. So what are the solutions that we have to attract the talent beyond the mission? We know
Google and Verily has set up a fund to subsidize hospitals paying for their AI and their machine learning people, right?
Did we decide that just a moment ago, David?
You hit a critically important point. You're right. Human capital is required for all of these things. And unfortunately, the draw of Silicon Valley has been tremendous on many of that capital. What I'm seeing now, which to me is one of the great positives of COVID-19, is people are leaning toward meaningful work. Whether it be working on a health project at Verily or Google or working at an MD Anderson is more attractive than working on selling a widget or figuring out which movie you're going to choose next. And I have seen in the last year when we would have positions, we would have one applicant. Now we have hundreds, all of whom are tremendous. And that's a big change that happened toward the end of COVID-19, that this new generation wants meaningful work or meaningful applications for their work.
I agree with that. We're absolutely seeing that people are seeking purpose and meaning in their work. And we see the same thing as people come to us because of Data Science Institute in oncology, that kind of work. And they are tired of writing algorithms to sell more socks or refrigerators. They want to unlock a lot of the mysteries that are hiding on inside our freezers and on our servers right now.
Exactly. And there's a point mentioned earlier today about more effective interactions with industry. And I think that academia can do that. Having come from academia and now in industry, there's a lot of opportunities. And in order for us to scale academic discovery, we're going to have to be in industry. So, so I think trying to get that continuum more set will help get us more training, because it's a critical need that you just mentioned.
Lily, as you look at that, and that was such an excellent point that I completely agree with, the partnership between academic institutions and industry as an industry person, how do you see that right now, and what are the ways in which you could choose an academic partner?
I think, so well, in my old role, we worked with academic partners all the time. I think, one of the things that when you're trying to do a research project in particular is that if you're not working with the end users and the clinicians and their patients, you're going to run off in the wrong direction pretty easily. And it's a huge waste of time and those large salaries, I suppose. Because you solve the wrong problem, right? Like healthcare is non-intuitive. And so that partnership between academia and medical centers and industry, I think is really critically important. I think another place that has been really great for us is as we are partnering with people, it's actually, there are folks that we can actually train and help gain the new skills, I would say, to do a lot of the things that the computer scientists and the other folks here can do. You don't need a Google computer scientist to train TensorFlow models, right? That is TensorFlow, and as well as other competitors, are open source. They're high school students that can actually train the models. So the model training isn't actually the hard part anymore. It's the data underneath and the labels underneath, right? And so if we can get that piece right, all the other things become much easier, and you'll have a lot more people that can help you with that. But the data piece, like I joke that like 80% of my old team, and actually probably my new team, is cleaning up the data, right? Because it's just in these, like we get them from hard drives, they get mailed in and it doesn't make sense. So I think that's the big piece where if we reduce the amount of almost manual labor that is required to implement technologies, we just make it easier for everyone. And that iteration speed will compound if we can get that infrastructure piece right. So I think it's not as hard as we think it is to get people who can build AI. It's way harder to get the data right.
No, I think you're totally right. I mean, what's wild is this happened before, right? Is that people who worked in the lab and the clinic never talked to each other. Then all of a sudden, translational programs that happened at hospitals like MD Anderson and others, and we were able to get things very quickly from the lab to the clinic. Now, the people who collect the data and industry need to be one team. We're all fighting the same freaking disease. So it is nonsense that they're not on the same Zoom calls in today's world, not talking together. Did the sun just set where you are or something? Or you were too still and the lights went off? But this is the new era. And I think we have to get over this notion that there's the dark side, which is industry, and there's the academic side. We're all one. And, without industry, you have no new drugs or treatment for disease. Without academics and hospitals, you have no samples and data of which to develop them. So the two are necessary for each other to succeed.
Such a great point, because we know that you need the clinicians on the front lines to define the use cases, as opposed to industry to work in a vacuum, in a silo, and present the solutions. That was the problem with IBM Watson. That was the solution, trying to find a problem, and it didn't work for a lot of good reasons, because people in the clinic were never involved at the inception and in the design. And if we can find ways to deepen the connection and refine the use cases and solve for the problems that the patients are experiencing, we'll all be a lot better, and we'll move faster, farther, quicker. To come back to the data dependency and the talent dependency, these are two really important points, because we have poor quality data. We have what David describes as bags of words. We have data scientists that come to institutions and spend 80% of their time cleaning up the data and 20% of their time analyzing the data. We have a lot of work to do on the quality of data or the data supply chain, so that we can give data scientists clean data all the time and reverse the time allocation that they spend at the moment. Put them 80% of the time on analyzing the data and 20% of the time cleaning it up. On the talent dependency, I'm just wondering from the two of you, David and Lily, if we need to go down to medical school training, where we don't really have tracks, and when bioinformatics is an old field that isn't quite what we're talking about today, and where we have to inspire young people who are thinking about a career of medicine and then marry data science in the ways in which we had biomedical science fused with medical training. Is that an opportunity that we're missing out on right now in medical schools, David?
Again, 2 words, hell yes. No question about it. You know, my son is a first-year medical student, and you know, I look and it's almost embarrassing watching. Don't tell him I said this, but you know, they're learning to memorize. He's doing infectious disease now, and he's memorizing every bug and antibiotics. You've got this little device where he can Google that, and then you know, use his brain for processing things and thinking about things and learning bioinformatics better, learning statistics, learning how to structure data. That's where we need to put the emphasis. We teach toward tests. We have these things called boards. Medical schools get graded on how well their students do on the boards, which has nothing to do with taking care of patients. Do you want a doctor who can memorize things really well, or do you want a doctor who can talk, is empathetic, and can think and process? I'd certainly rather the latter.
Well, we could get onto a tangent or a pathway here that's super important, but I'm completely aligned with your thinking. We have old school methodology anchored in the last millennium that's based on organic chemistry and MCAT testing to bring people into medical school. We do not screen for compassion and empathy or communication skills. If we had emotional intelligence as a criterion that we selected for, you would get a totally different kind of medical student. And if you accepted the reality, everything that medical students need to know, which used to be in textbooks and required memorization, is now on an iPhone. And you simply need to know how to curate and cultivate information that you can pull up on an iPhone and test people for the ability to explain that in plain language to patients. That's really a totally different way of thinking about medical education. And the thing that's holding us back is exactly what you pointed out. It's test scores in U.S. News and World Report ranking.
You know, I do autopsies on all my patients who pass away from cancer. And I learned a tremendous amount. I have a feedback loop, right? I thought it was scar tissue in the lung. It turned out to be cancer. I couldn't understand why there was short of breath, and there's a single layer of cancer cells circulating in their alveoli in their lung. They can't get oxygen in. I learned so much from it, but we've lost all those things, right? We've moved to the pure knowledge where we just want to say, what is the answer? How can I Google it and get to that? We need to go back to the art of it and learn it. And then you need to understand what that data really is. You know, we don't have standards for the data. Lily calls it a fractured leg. I call it a broken leg. So no matter how much we try to harmonize our database, they may be a little bit off.
And going back to the education, I think how we communicate with patients and the need for specific communications, like we heard from Rob Wynn. I mean, we have to be able to teach our students the empathy, getting trusted messengers into the right communities, to educate about new technologies. And because if we're going to implement these new technologies, we need to really have those discussions about what are the pros, what are the cons, and how we can see that this is for the greater good. And I think we're getting that's being lost with the test scores and the rankings, no question.
One other area that I want to drill down into relates to the use of algorithms in clinical medicine. And one of the areas, of course, that we've looked at is the identification of patients who have sepsis in the hospital. And as these algorithms have been deployed, we've uncovered a new field that's referred to as algorithm bias, where algorithms that are developed in one institution or by one group of researchers on one small subset of patients don't appear to work very well when they're applied in other settings. Lily, what are your thoughts on how we can avoid that? Because it has a lot of implications to how we develop the algorithms.
Yeah, absolutely. And I hate to sound like a broken record, but it's real world data in connected systems. I think, there's this concept of like post-market monitoring, right? And for devices and algorithms are sort of basically software as a medical device, right? There's opportunity to actually be looking at how they perform even after approval. And these are all, like, these devices actually they're served maybe through a cloud-based, but they're connected. They can be connected, and you can actually get real-time or near real-time analysis on how things are going. So instead of relying on an adverse event, like a post-market surveillance, which is when an adverse event happens and then you report and then it takes forever and then you respond, you can actually see hotspots where there are deviations from performance, right? So I think there's to answer the question actually more comprehensively, there are multiple ways of dealing with bias. The first one is before you even build a product, right? You're actually doing the work in the communities where you want to deploy this algorithm. Your training data represents a wide variety of participants and what they go through to actually access care. And so from the design aspect, you can actually prevent some of the stuff from happening. Also on the design side, you can actually start looking out for where to look, right? Once the algorithm's deployed, if you're going to monitor, you actually have to know where to look. You can't search for everything. There's no such thing, right? And so if you notice that, hey, older folks tend to struggle or have worse results with this algorithm, you actually put that in the design of the product and you say, post-market, we're going to be watching this one very carefully, right? And in fact, every time we go to a new site, we go live there, we're going to increase monitoring, and then so that we can respond if we need to, right? So when you want to get that post-market and the continuous monitoring right, you have to design for it from the beginning. So I think we have to think about it as a continuum, but that last piece about looking at how a product actually performs in the real world when it's used off-label on a different population that it was originally intended. That is what happens in real life, and we need data on that right now, and we shouldn't ignore that data. So I think it's really great, and I think the problem though is how do we incentivize manufacturers like industry to do that, right? Because I think the great part of some of this stuff is that if you're able to get that data about off-label use, you could maybe expand your indications, right? So there are win-win situations here. We just need to prove those cases out and make it clear what the win-win is for everyone in the situation. Obviously, the patient, as a main stakeholder, but also everyone else that's involved.
I agree with everything you said, Lily. The one thing I would add is just transparency on the root data. That it's very important that you're very transparent to what the data was you made your algorithm or your decision upon, so others can say, does my patient fit that data set?
Yes, absolutely. There are these things called model cards. Sorry to interrupt, but they're model cards. It's kind of the nutrition facts of the model, right? Like, what went in there so that you can also make decisions there. So that's a really good point.
And the final thing, I agree with everything. The final thing I would add would be simplicity. So when you're getting the post-market data, how can you get the data easily, simply, without clinical trials or this? How do we get that in? I think that's a challenge for us, being able to get that and have it be regulatory grade.
Great points, superb discussion. Let's go to the audience for audience questions now.
Good afternoon. Chris Stewart, a retired social behavioral scientist, and have dealt with many of these same issues on DARPA programs of cleaning, munging data, and the limitations, the fundamental limitations that we have in this area because of not being able to use common data sets. And I guess one of the questions is, and I was at a National Academy session, 4 or 5 years ago, and really eye-opening when we were talking about the genetic testing and obviously the costs going down, but it wasn't really until we hit 700,000 cases that we began to see the right kinds of patterns to begin to understand the genetic code. Now we realized long read sequencing is more important, or is valuable. We've also realized junk DNA is important to look at in between and the implications there. So I guess one of the questions is, how are we going to do the power analysis? How are we going to get to the numbers to crack the code that we need to do? because I know from a trust framework standpoint that patients aren't going to be willing to share their data in the volumes that we want. And I worked on dynamic consent, I've worked on those sorts of issues, and it is a wicked problem.
Terrific question. I guess I'll go, Lily, to you for the thoughts on this. How do we get to critical mass with high quality data so we can get to exit velocity here?
Oh, such a good question and such a hard problem. And thank you for asking it. I think, and thank you for, I think there are two ways that I would think about this. The first one is, how do we deliver value to patients, participants, in such a way that they will want to do this? The reason I think about this is, confession, I play Pokemon Go. I still play Pokemon Go. I know we did it in 2016, it's kind of thing in the past. They have 100 million people engaged playing this game, and you run around a city with your face buried in front. Have you ever played in this? And you look kind of ridiculous, but there are adults playing this. I say I play with my kids, but there are adults playing this, and there is a value that is exchanged, right? And so we do have lots of different apps and other things that engage users or give value back to users such that they will want to do certain things. And so I think, I don't know we have the answer, but there are other industries that we can learn from about how to give users value in such a way that they will want to engage. I think that's first thing. And also recognizing that people aren't coin operated. It's not just about giving people money to do the thing, right? Like, I don't get money to play Pokemon. In fact, I pay money to do this thing, right? So it's not just about money, it's about other things. And so we have to think about value broadly. So that's one piece. The other piece is interoperability, right? So we've got a few hundred folks who are excited to do stuff here. We got another few hundred folks that are, here. And if you aggregate it over, you aggregate over multiple academic centers, multiple manufacturers, you actually have you may have lots of data. They just are lots of people that are interested in, right? Interested in helping, but they just have all these different kinds of data, and we can't make them work together, right? We have people like Army of Women. We have women who are willing, hundreds of thousands of women who are willing to, normal women that are literally willing to get biopsies, right, to help with something like this. So I can't imagine I think we can activate the people, but when we do activate folks, we have to make good on the promise that their donation is going to multiply beyond one single study. So I think those are the two ways that I would try to address the problem, but I acknowledge this is a big problem and we have to figure it out.
Okay, I reduced that to gamification and altruism. Okay, next question.
Dr. Agus, to your point, Yasit Virisuri, I represent an institution that trains a lot of nurses, wrote memorization and board exams. The National Board on National Council on State Boards of Nursing is revising its assessment next year for the national boards and testing the high levels of cognitive testing and clinical judgment, and essentially just changing the way they test. It's going to be a milestone change. But you treated Steve Jobs. First question, how was it to treat him? And number two, you have some thoughts about supplements and their impact on cancer. Would you comment?
David, over to you. Hold on.
To date, there has yet to be a study showing a benefit to a vitamin or a supplement in a normal individual in this world. And so certainly I'm not a believer in taking things without data. We are a complex emergent system. So the notion in a reductionist way, take this will happen, doesn't make a lot of sense. Steve was a remarkable man. I think, I'll end here because I think it's a very important note, is that, he said to me once, why can't you look at my cancer that I like it a code, and where there's an error in the code, I can correct it, and then everything is okay. And I think it's a very prescient thing is to try to, get reductionist in cancer. We tried to do with genomics, and it got us a little bit ways there. We have some certainly molecular targeted therapies that have had benefit, but it has not solved the problem of cancer. We have a long way to go, and it is a complex emergent system. And so the way to get to complex emergent systems is to have large data sets. And you don't need an understanding necessarily of all the biology in order to make an intervention that has a benefit. And I think that's the way we're going to go.
I think we have time for one more question. Anna, you get the last question.
Well, this may not go over well, but I'll ask it anyway. So, first of all,
I expect that of you.
Yes, of course you do. So, with all due respect, I want to ask Bill a question about this essay that is so infamous now. It's on everybody's mind. And I will say this before I ask my question. In my mind, we have invested enormous amounts of our resources in creating molecular data sets and wildly popular cellular assays, et cetera. Having said that, most of that work has gone toward identifying new targets for therapy. And so I think we should be thinking about how does that apply to prevent cancer, and how is that going to apply to detect disease very early. So that's all for saying that I think this is a good idea, and I think it will play out as a good idea. But the question I have for you is, why are you doing this as a laboratory-developed test and selling it before you had the data to actually know? And I'm asking the question based on the fact that laboratory-developed tests will be regulated very soon, I think, by the FDA. The VALID Act is being actually talked about today in Congress, as a matter of fact. And I just wonder why Grail didn't go to the trouble of actually pursuing this test as a PMA from day one. Why did you choose to sell it under CLIA versus going after the approval for FDA?
So
I told you wouldn't like the question. So.
No, and it's a good question. I don't have all the answers for you in the few months I've been at Grail. But what I can tell you is that this test is picking up cancers that are making a difference now. My eureka moment was a 2-centimeter body of pancreas cancer. As a pancreatic cancer surgeon, I've never seen that. So we are making a difference in local control if we're not making a difference in survival. A 2-centimeter hypopharyngeal cancer, the Mayo Clinic called about that. They go, we've never seen it. What do we do? Do we need to radiate? What do we do? So I think there's efficacy that we've been seeing. The actual the decision between LDT going straight for PMA, was, I don't know all the details why that decision was made, but seeing the efficacy and seeing it in so many thousands of patients, hundreds of thousands of patients, or implementing it in hundreds of thousands of patients was our logic. Sure, and the question was, for those who weren't on microphone, is this like the shades of Elizabeth Holmes, et cetera? And we get asked that a lot. This is not a drop of blood without a lot of data. This is very, very deep science that I believe, and I believe that the technology is going to spawn a lot more than just early detection, because we haven't looked at regulation. We've focused on DNA and DNA mutations, and that's a fraction of the problem. It's regulation. And I believe that those methylation profiles are going to identify subsets of high-risk people down the road, probably predict who responds to therapy for immunotherapy. So I think that's the broad spectrum of this. So our broad, deep approach to over 400,000 tests so far in 330,000 clinical trials, I think is a very deep approach that does not that is far different from the other approaches you described.
I want to just acknowledge the richness of the discussion and thank our panel. It's just been a terrific panel. We could have gone on for 2 more hours. And you can see by the depth of the issues and the things that we were just scratching the surface of. This is a super talented panel. I want to thank David, Lily, and Bill for their talent, their time, their thoughts. Thank you all so much.
Thank you.
Ladies and gentlemen, let's thank all of our program participants again over the last 2 days. Thank you. What a great way to conclude. I could have listened for another 2 hours. I think just concluding with the emphasis on the future, it kind of ties back to what Dr. von Eschenbach was saying yesterday, using the past to influence the future, and that's the point of this conference and really what this is all about. So I hope that you all found this beneficial, and now I want to invite everyone to join Dr. Will Lee in the gift shop, where he will be signing copies of his book, How to Eat to Beat Disease. I also invite you all to follow the Nixon Foundation across social media, our handle @Nixon Foundation across all social media. Thank you all for attending, and we'll see you back here next year in 2023. Thank you.